edition here. We are really delighted to have all of you here, and we look forward to a wonderful 3-day interaction. Hope all of you have a pleasant stay in Allahabad and have a great learning experience interacting with your colleagues and others in the conference. Just about the conference, so we received 214 abstracts this time, and after the peer review, 90 submissions were accepted for poster and 29 for oral presentations. Finally, we are having 72 posters in these 3 days, 27 oral 27 talks, and 4 keynotes. We will now begin with the first keynote by Professor Chandan Vaidya, Georgetown University. And we have a formal inaugural after some time, after the first keynote. So it's a pleasure to have Professor Vaidya. She has been here a couple of times earlier. And as we all know, she's a developmental cognitive neuroscientist. And her research focuses on characterizing the functional neural architecture of adaptive mechanisms during the lifespan, as well as developmental disorders like ADHD and autism spectrum disorder. It's my pleasure to invite her to deliver her talk on adaptive functioning in neurodevelopmental disorders. And it's also very pleasant to invite Professor Narayanan Srinivasan as the chair of this session. So over to you, Professor Narayanan. All of you know Narayanan, so I don't think I have to introduce him, but he's a professor at IIT Kanpur. Formerly, he was here. He has been our colleague for 15 years, so it's really nice to have him back, and he'll be chairing the session. Thank you.
Yeah, thanks, Bhumika. I think there was already some introduction, but let me add some more. Professor Chandan Vaidya did her undergraduate degree at Sophia College, University of Mumbai. Got her PhD from Syracuse University and has been at Georgetown for, I think, a long time. So she works on adaptive mechanisms throughout the lifespan, and I think she's... Yours is loaded? Yeah. Oh, sorry. Okay, fine. So, may I invite Professor Chandan Vaidya for a keynote address. Thank you.
I'll just use this.
Okay.
Yeah, I'll use the keyboard actually.
Awesome. Okay, ma'am.
Yeah, it's much better. Good morning, everyone. Thank you so much for that kind introduction, and I'm really thrilled to be here. I had the privilege of being invited to the first one, the first of this society. So it's really a thrill to be here 12 years later, and then the importance of the 12th right here is also a special thing. So anyway, thank you all very much for this invitation, and I'm happy to talk about some of our recent work and also to see, so many trainees here. Happy to talk to you all, so don't be shy. Come and talk and tell us, all of us speakers, what your research is about. So, water is falling here. So to just get started, that's the view from my office right now. So I'm very happy to be here and not there, although it only looks pretty from far. So just to get started, adaptive function is essentially adjusting to real life, right? So when we think about what is the primary goal of development, it is that the person at the end of development or during development adjusts to their environment, right? They're able to make good decisions. They're able to solve problems that come their way. They can interact with their peers or anyone socially within the family or the community. communicate well with them, and do their daily practical activities, like dress yourself and get yourself to work, for example, or to school, right? So that's what we mean by adaptive functions. And when you think about it, anyone presenting with a psychiatric problem, by definition, fails in some of these activities, and that is what triggers the individual to seek treatment, right? And so it's really an important kind of end goal or an outcome of development, and also a target of intervention efforts, right, in order to get the person back into the community. However, people differ. There are sources of variation. Certainly in any psychiatric condition, there's failures in some of these, and people vary along those dimensions. And finding what the key processes are to intervene is an important goal overall, right? So that's the purpose. So I want to just illustrate just an example. This is from an old study that we did, just to drive home the point and pinpoint some of the processes that may be interesting for us to focus on. So I'm just going to do a quick exercise, and this is from our experiment that we did just with college students, okay? So we're not talking about disorders as yet. So you can play along. So your job here is to decide if the last statement in the logical syllogism that I'm going to put up here is valid or not, right? Very simple. Making good decisions depends upon good logical reasoning abilities, and so this is a reason I want to kind of illustrate this, right? So just play along. Here's the first one, very simple. This is, your job is to validate the last statement. So children are older than infants, given the first two statements, right? Does it logically follow? So this is a simple one, and hopefully the answer is yes, right? So this is a valid statement following logically from the first two. Here's another one. Can you guys see? Yeah? Yeah. So cockroaches are smaller than snakes. Cockroaches are bigger than maggots. Snakes are bigger than maggots, right? Valid. The subject material is a little icky, right? Valid statement. Here comes another one. Trees are taller than flowers. Trees are shorter than grass. Flowers are shorter than grass. Is the last statement logically valid? Yes, it is. But it tripped you a little bit, right? Because flowers are not shorter than grass in real life, right? And so you kind of have to suppress that belief and just remind yourself that this is a logic, I need to stick with the logic, right? So here comes another one. Tobacco is more poisonous than venom. Tobacco is less poisonous than mucus. Venom is less poisonous than mucus. Okay, not true in real life, but valid logically. And the subject matter is icky. It's emotional in some sense, right? And so you could see now where I'm going with this. So what we did here is that we manipulated the emotional content and the congruence with our real life beliefs. And interestingly, what you find is that this type of susceptibility to be led away, to be swayed by your real life beliefs varies across people. Okay, so not everyone is equally susceptible. And what we found is that trait anxiety actually had an interaction such that those with higher trait anxiety were more susceptible to the belief bias for the emotional content, right? Quite selectively. And further along, this is part of Melanie Stolstorff's dissertation a long time ago, this proneness to anxiety is biologically rooted, partially at least, right? So if you carry the short allele of the serotonin transporter genetic polymorphism, you are more prone to anxiety. So this is a temperamental trait. And so we found this very selective effect such that those who carried the short allele were much more susceptible to this belief bias, right? So what I'm trying to illustrate here is that these adaptive abilities, are quite importantly rooted in biology. And here, the process we were going after is a component of executive functioning, which we'll get to, right? The ability to suppress irrelevant information, in this case, our own beliefs, right, about the real world. So the big question here is that adaptive functioning is affected in all psychiatric conditions. We're going to be focused on pediatric conditions here. And they're the target of intervention efforts. And our big question is, what are these mediating processes, right? Let's find them. Let's study them in the brain, and so we can intervene appropriately. And, oops, what happened here? Did I touch something? It's okay. Okay, good. Does this lock? Yeah. Okay, perfect. Okay, I'll do this, yeah. Thank you. It's easier for me to do this, yeah. All right, so let's focus on executive functioning. Here I showed you an interaction with emotion, right? And again, I think many of you may be familiar with this. Many of you might be familiar with executive functioning, but just very quickly, it's an umbrella term that captures a variety of component processes. Some of the important ones are working memory, cognitive set shifting, mental set shifting, which goes to flexibility, inhibitory control, which is what we illustrated here, as well as a variety of other types of skill sets. For example, ability to plan, to organize, and so on, right? So it's a big bag of a variety of things. But critically important, this is one of my very favorite studies and data sets out there. Again, maybe some of you know about this one. It's a very landmark longitudinal study that has been going on for now 50 years. So the cohort now is, I think, 52 presently. And they've been followed from really literally from birth. And what they found, and this is in a particular town in New Zealand, so very homogeneous cohort, so that you can control a variety of things. And what they found is that very early, so between ages 3 to 11, executive functioning early on has very profound impacts on predicting adult outcomes in a variety of things. So physical health, substance use, substance abuse, financial status, where you end up financially, as well as rates of incarceration and so on, above and beyond other things like IQ, socioeconomic status early on, right? And if you look at what is being measured, it's a variety of very classic symptoms that one would measure for things like attention deficit hyperactivity disorder, but overall, it's really basic components of executive functioning: impulsivity, hyperactivity, lack of persistence, inattention, right? So these have very profound impacts way later, right? At age, in this case, age 32. That is really, really far away. It's really important for us to try to delineate and discover what these sources of variation are in terms of executive functioning, what are the component processes that are critical, as well as look at their neural correlates. But it's complex, and that's a problem. Executive functioning is multidimensional. Tasks are not process pure, Some of you may recognize this model from Friedman and Miyake. And what it shows is that all of these things we measure as executive functioning have a lot of common variance, right? It's hard to truly isolate these processes. In fact, the best fitting model is this bifactor model that shows sort of a common EF factor and then some of these others like set shifting and working memory and updating, right? So that's a problem in terms of if we really want to pinpoint what to intervene on and so on. And critically, it's messy because it interacts with psychopathology in a very complex way, such that, so let's just take ADHD, which is the quintessential disorder, let's say, of executive functioning, right? So all of these things we think about as failures of executive functioning, we would typically be measuring for diagnosing ADHD. For sort of short form of that is distractibility, hyperactivity, impulsivity, and if one has these symptoms or difficulties in these areas, what you find is that it raises the risk for other types of psychopathology. Okay, so if there presence of ADHD symptoms, presence of executive function is related, fourfold higher chance of an internalizing condition, which is a mood disorder, anxiety, depression, tenfold higher risk for a externalizing condition, which is the acting out, aggression, delinquency, and all that, right? So the point here is that this is all very highly meshed together. There's very high comorbidity, sort of trying to isolate what is an executive problem outside of these other emotional issues is very difficult, right? So we have to sort of really consider that as a whole together. Autism, you might think, is quite distinct because, it has very specific diagnostic criteria in terms of social problems, in terms of repetitive behavior, but the presence of executive dysfunction is extremely high. Over 70% of people with autism will have some executive problems, if not meet the diagnostic criteria for ADHD. And what that does is that it leads to really bad adaptive functioning. You can fix some of the social issues, some of the core symptoms of autism, but the adaptive functioning doesn't improve, right? So you need that executive piece to be intervened on in order to get the adaptive benefits. But this is a problem, right? It's a huge, this elephant that is so kind of multidimensional, given the psychopathology, given the complexity of executive functioning, we need to capture. And so one approach has been, both our lab as well as others, is to take this transdiagnostic approach, right? So let's take large samples of kids or people where you are kind of capture the whole mess of it, right? So all of that comorbidity, all of that concurrent symptoms, and then let's try to pull out what may be common, right? Let's not worry so much about the diagnostic categories because to some extent they're meaningless given all of the comorbidity. So what I'm going to be describing are two studies we've done that kind of take a stab at this, and they're a little bit distinct from each other, so I will get into that. Overall, these are all samples that are 7 to 13 years. They have a variety of diagnoses that are, common in childhood, except for medical diagnoses, so no epilepsy, no tumors and things like that. But they're all presenting to clinics with a behavioral problem, so that's our inclusion. And then we control for age and gender and IQ. So let's take the first one. This is Adam Kaminsky, my PhD student. Now he's a postdoc, but part of his thesis work. And what we did here is that we looked at psychopathology in a different way than at least we had done before. So when you look at a variety of symptoms or measures of any kind of psychopathology, what you again see is that there's high covariance, right? And so even internalizing, externalizing, right? We think of them as two different things in terms of anxiety, depression versus aggression, acting out. There's common variance, and that you can capture with, factor analytic techniques or principal component analyses, and you get sort of this general psychopathology factor, right? Which is referred to as the P factor, which accounts for about half the variance in that, right? So it's useful to capture something common, and so that's what we did here. And then we asked, we know that general, psychopathology, and I'll show you data, so that psychopathology relates to poor adaptive functioning. So that association is well established. And so what we want to discover are sort of the mediating processes, right, of that association. Perhaps partially mediating, but nonetheless, that's the question we were asking. So here's the data. We used principal component analyses for the child behavior checklist, a very common clinical instrument. Again, it accounts for a P factor for about, like I said, half, 54% of the variance. And you can see here that higher general psychopathology, which is here, relates to worse adaptive functioning, right? So that's the association we want to explain. And then in the brain, we designed this task, and I'm going to show you the task a little bit, trying to really capture a variety of the elements, right? So we wanted sort of the meaningful, socially important stimuli, so we used faces. And we wanted to capture the dynamic aspects of executive functioning, and so here's the task. All you have to do is to press an arrow for the direction of an arrow that's going to be on a face. You'll see whether it's pointing left or right. It's a very simple task. Hopefully you can see this. Let's see. See the arrow that is in the center? Okay, right, so you got the task. And so what it does is that the eye gaze is either congruent or incongruent, right? This is our classic kind of Stroop effect, but we vary the sequence such that you can capture what is a very classic effect in cognitive psychology called the conflict adaptation effect, right? So the idea is that when you have an incongruent trial, which is going to be harder, so slower, more error prone, the trial that came before that matters. If it was congruent, that current incongruent trial is going to be harder, right? But the successive incongruent trial actually gets easier, right? So the idea is that you've stepped up control and now you're there, right? And so the next incongruent trial, you can apply it, right? And so you get this very classic effect that is captured in reaction time or mean accuracy. These were kids, and so accuracy works well. And so here it is that when the current trial follows an incongruent trial, you're more accurate than if it had been a congruent trial, right? So it's a very nice effect. And so we're looking in the brain here during this task, and we used a seed-based functional connectivity analyses. This is a psychophysiological interaction for those of you who do that work. With this dorsal anterior cingulate seed, okay? And so we did this, Adam did this very carefully because that area, this patch, is actually parcellated in such a way that actually it belongs to multiple networks, right? So a component of that is part of the frontoparietal network, a component of that is part of the salience network, and so with this, we did this carefully, and what we found is that connections with middle frontal gyrus, right hemisphere, and parietal cortex, shown here, for the frontoparietal seed per se, mediated partially this association between the general P factor and adaptive functioning, right? So this is a critical connection. Theoretically, we would have probably predicted that. We know that the anterior cingulate and these connections are important for managing conflict and sort of the dynamic up and down of, stepping up control and stepping down control, which is sort of a proxy, for real life. And so here we have I think, a nice finding. So this is one of the things I would say is a mediating process that we found, and then one could target interventions in terms of trying to capture sort of this flexibility of upping and modulating cognitive control. In the second approach, we sort of took on this challenge of the heterogeneity of executive functioning, right? So remember, we are dealing with a transdiagnostic sample here, and we wanted to first ask, is executive functioning sort of the same in terms of, its failures across all these people, right? And the simple answer is that it shouldn't be. Clinicians know that not every child with ADHD or autism or executive problem has the same type of problem, right? Some are more impulsive, for others it's more organization, planning, and so on. So we wanted to kind of capture that and sort of see what that structure may be. And so we used a data-driven approach. So these are the questions we're asking, are there qualitatively different executive function profiles that we could discover in these, using sort of a large data set, and then what does it look in the brain, right? Are they distinguished in the brain? And again, this big question, what this type of approach is asking, is that we need just to know not only what to treat, but who to treat, right? So the person question is an important one here. And so the methods here we used, this is my postdoc at the time, Dr. Yu's work. We used community detection as an approach, applied it to 2 separate samples. You can see here, we collaborated with Kennedy Krieger. Stuart Mostowski had a data set that was well matched in terms of age and all of that. And here we had 80 kids who had diagnosis of ADHD, kids who had diagnosis of autism, and typical development, right? So these were three subsamples that we had within that. And what we did was we used the Louvain algorithm to look for homogeneous communities. What this method does is that it's looking for correlations across people for the features that you're inputting. And the features that we use are the basic clinical scores that typically any clinic would have, right, for children. So you have the ADHD rating scale. The brief, some of you know this, has 8 domains of executive functioning, and then the internalizing, externalizing indices that we talked about. So we had these 12, and we applied it to that. And we did a variety of validation, train on one sample, test on the other. So you see the accuracies there, split half, validation across the whole thing. And what we found is this. We found 3 profiles, EF profiles as we're calling them. I've color-coded them there, so just follow along with me. What you have here are the 12 measures going from ADHD, rating scale, hyperactivity, inattention, across the brief, and then the externalizing, internalizing at the end here, right? So let's take the first one, the red one. We were calling it the flex EMOT for flexibility emotional problems. Peaks are worse scores, so those are the problems, right? The troughs, the lower, are less of a problem, right? So think of these as profiles of strengths and weaknesses. And so you can see very clearly, the red one, the primary issues are with shifting and emotional control, right? And then you see internalizing also high up there, right? Both externalizing, internalizing, but it kind of goes with the emotional piece, right? So this is a subgroup that really has the flexibility and emotional regulation as their main issue. Look at the green one. These, you may say, are sort of what we think of as classic ADHD, perhaps. Very primarily, hyperactivity inhibition is the problem, but nothing, lower, okay, maybe there's a little bit of a problem, but not really one that you would select for intervening. And the emotional piece is pretty low here, right? And then the third one is a very classic working memory planning organization issue, as you can see here with all of these. And then not so much emotional, right? So and we called it metacog because the brief calls this cluster as the metacognition index, right? So that's why we called it that. So you see, these are sort of three relatively distinct qualitative problems which would be useful, right? So we had the support vector machine we built. You could input these 12 scores and then get which profile the person belongs, the child belongs to, and then it sort of makes the work a little bit easier. So no differences by age group and IQ and so on. In the brain, we happen to have for 84 of these kids, and the big thing is, of course, this cuts across diagnosis, right? So ADHD, autism, but also typically development. I didn't show you that graph just because of time, but it's in the paper. So if you just pull the typically developing and do this exact thing, you get the same exact structure, which tells you that psychopathology is kind of nested, right, within sort of normal temperamental traits, right? And so any developmental psychologist already knew that, but it's kind of nice to see that. So in the brain, I had data on a selective attention task on about 84 of these kids. And so we looked at that and we just asked a very simple question in terms of activation during that task, whether the DSM-based MANOVA, accounts for more variance or the EF profile-based one, right? So it's very, very straightforward. And what we find is that the EF profile one was better in terms of, significant in terms of accounting for variance. And you can see the activation, very classic attentional regions, right? So you have, frontoparietal, cingulates, all of the salience network, dorsal attention, and so on. And so this is, just a neurobiological validation for these profiles. What we're doing now, and we're currently getting ready to submit this, is a replication, right? I mean, remember, data-driven work, it depends on your cohort, right? So what you discover is very much dependent on who you put in there in terms of people. And so we wanted to see whether these profiles are generalizable to, another data set. So this is now a new data set, 1,472, and enriched for psychopathology. Okay, so this now is a messier group, not just ADHD, not just autism, name it, any behavioral problem, even things like dyslexia. The only exclusion is that they don't have any medical issues, right? Epilepsy, tumors, Fragile X, genetics, things like that. And so what we found is that the first two, the flexibility, emotional problem profile, and the metacog, the working memory one, perfectly replicates. Okay, we got the same exact thing. But the inhibit one, the one that had this selective inhibition problem, we couldn't replicate, it didn't. And in fact, what we find is that it's something more complex, so that it's kids who have peaks on all of the things, right? So inhibition, shifting, as well as working memory, that sort of cluster together, right? And so these are kids who have overall more severe psychopathology, and so they were clustered as the third one. So that's been important to know, right? So the sample matters, right, and the amount of psychopathology. What we did for the brain here is that we capitalized on this notion of a general EF factor, right? So remember, I said all these measures are not process pure. There's a lot of common variance. And so all of the kids, not the 1,000-something, this is a sample of 92, I think, that we had, so this was a prospective study that went through an NBAC task for working memory, that same dynamic social stroop, and the stop signal, right? Very classic inhibitory task. But we were interested in sort of capturing the general EF, and so we're simply using the concatenated time series of all of the whole thing for every person, right? And then we are doing whole brain functional connectivity, parcel-wise, so it's sort of like a searchlight approach. So 388 parcels, looking at every parcel across the brain, right? And then what we're doing is to see what predicts both the EF subgroups, right, and adaptive functioning. And so the parents filled out a scale for their real life functioning that is a standardized scale. And so when we do that, what we find is that it's a very distributed network, right? So 26 regions, those of you who know the network out there, these are the 7 Yo networks. And what we find is that parcels from all of these networks are predictive of both of these things, right? So the point here is that it's a distributed system that makes sense. And so connectivity of highly distributed regions, including, we were surprised to see the somatomotor network come up so much. So if you look here, there's 9 parcels from the somatomotor network. And perhaps it makes sense given that these are all tasks where you have to plan responses and so on. That are predictive, right? So anyway, so that's, I'll leave this there, but this is sort of has been one approach where we're trying to include adaptive functioning in the kinds of things we are looking at so that it has some relevance for, outcomes of development, right? So those are, just summing up here, these are sort of the 2 things that we looked at here. So moving on. So we've so far been talking about executive functioning, right? And we've talked about sort of really taking this very granular approach to a very specific connection, right? Dorsal cingulate related. Then we took this whole brain approach to sort of looking at all sort of the distributed networks. But it's not the only way we adapt to the environment, right? So the executive functioning is sort of an effortful way, but the effortless way is learning, right? So we very spontaneously learn about the environment. We abstract out commonalities across experiences and then are able to apply them to new situations, right? In fact, that's a fundamental theoretical way in terms of, what should promote flexibility, right? And so what we've started to look at now is generalization, so within a category learning paradigm. So where you get trained, you acquire information during a training episode with feedback. So you don't know what you're learning, but feedback-based learning, and then we test on new stimuli. Whether they've learned category structure, right? And then we look at, its links to adaptive functioning and so on. So let me tell you a little bit about these. This is more ongoing work, not published, but again, we have one paper that is under review, so I'm going to share that with you, right? So the goal here is we are looking specifically at the role of abstract representation during category learning. And it's our first foray into doing computational model-based fMRI, right? So there are models out there that are similarity-based models. Some of you may do this work. I'm going to go through this. It's busy. But let's just focus on C for a moment. And so what you can model is the trial-by-trial generalization performance. And you can look at similarity, model similarity based on a prototype that was the origin of the stimuli that the person is seeing. So our stimuli set here, one of them is illustrated. So we manipulate the features away from the prototype very systematically. So 1 feature away, 2 feature away, 3 features away. And then what they do is that they do a out-of-scanner training task, and we train them with 2 away stimuli. And they get, 2 fictional families that these fish belong to, go with your gut, and they'll press one button or the other, correct or not, and then over time, I think it's like 300 or so trials, 5 blocks, you get a very nice accuracy increase. And these are kids who are 14 to 17 years old. We particularly picked the teen range, and this is, by the way, done in autism, teach them, but cannot apply it to new things, right? Or have a less of an ability to apply it. So that's why we're looking at this. So out in the scanner, they get this full series, including the prototype, right? And then they have to decide, apply sort of which family they belong for, and you can look at the accuracy there. So that's, and then you model based on whether their performance aligns with similarity to a prototype or similarity to the trained exemplars, right? So that's the exemplar-based learning and prototype-based learning. So you'd get parameters for both of these. The brain can do both, right? But typically people have one dominant mechanism. We are basing all of this work on adult work, by the way, really beautiful work done by Topa, who's at the University of Oregon. And so this is, she's a collaborator on this, and so we created these new data sets, stimuli sets based on, all of her work. And so what we have here are 65 kids, like I said, 14 to 18-year-olds, all autistic, normal IQs. They were tested twice, 9 months apart. Because we want to be sure whether their dominance is actually stable, because unstable would be another indicator, right? In adulthood, it's supposed to be stable, right? In sort of typical development, so that's why we did. Then this is also part of a longer intervention study that I'll talk about in a minute. Anyway, so that's what we're doing. We apply these computational models, and then you get these parameters that you can apply to actually delineate the regressors that you can then use for analyzing your brain imaging data, right? So then, in the brain, you can know which regions are tracking the prototype and which regions are tracking the exemplar-based learning. And perhaps, for an individual, both are equally effective, right? Theoretically, the prototype mechanism is more effective because it's more efficient, right? And so on. In adults, you see majority of adults actually show dominance for a prototype. What we found, actually, behaviorally was surprising. In autism, we didn't expect this. We found that 2/3 of the sample actually had a prototype dominance as their learning mechanism. So this is actually really amazing news, because the theory says that it should be the weaker one in autism, right? But in autism, again, the critical thing is heterogeneity, right? If you've seen one case of autism, you've seen one case of autism, right? So this is one of the messages of the paper. It's under review now. This is my postdoc, Eugene Chen's work. The key thing here that I will share, because we are talking about adaptive functioning here, is what about the brain, right? So before I get to that, I want to prove that generalization has something to do with adaptive functioning. And we did, in fact, see that. So you see this correlation. And this is the ABAS that was done. Some of you may know this is a pediatric measurement for adaptive functioning. And we do see links with generalization accuracy, right? In the brain, we found one region where we see this interaction with the prototype-based neural correlate and the severity of symptoms in autism. So what we found here is that those the kids who are at the more severe end, which are shown here, this was just a median split on the sample. So these are the triangles are the severe. They show this nice relationship in terms of drawing on this left intraparietal region more in terms of prototype tracking for those who have better adaptive functioning, right? So this patch in parietal cortex has a lot to do with visual spatial representation. It's left hemisphere, so you have sort of the motor piece in there as well. So left parietal is important. So we think it's it's an abstract representation that perhaps has as the basis that type of information, right? So anyway, so far I'm telling you, we talked about executive functioning, talked a little bit about learning and how that may be important for adaptive functioning and specifically abstract representation. However, these two things are not really that separate, right? Theoretically, they ought to go hand in hand, and that's kind of what development in real life is about, that both of these probably feed into each other. It may be bidirectional, right? And so being able to do well abstraction across experiences helps you be flexible. Perhaps your just straight level flexibility helps you do abstraction, right? Well, it's probably both. But what we're trying to do then is to really take a look at this in the same cohort where after the first two data points that were sort of 8-9 months apart, they go into intervention for flexibility. And again, some of you may know this intervention. This is work with my colleague, longtime collaborator, Lauren Kenworthy, and her group, who have designed the Unstuck and On-Target intervention. That's at preschool and all the way to the teens. And so right now they're sort of fine-tuning the teen one, which is what we used here. It's a unique intervention because it goes through their school year. It's like an 8-month intervention that does a variety of things, right? So they're being very deliberately taught to think big picture, to have sort of options at the back in their back pocket, so to speak, in terms of working on that flexibility and planning and so on. And so it was run like a cognitive behavioral therapy model, so that the kids met every once a week for 8 months, basically, and all different. That's what's listed here in terms of the weeks here, 1 to 26. That they were working on certain things and they had certain products to produce and all that. So I don't have data yet. We are very actively looking at it. We do have all the imaging completed now for the third time point. And hopefully you'll invite me back and I'll share the data with you. So anyway, so the big question we're asking is that if you intervene on flexibility, do you change learning? Do you make it more deeper, stronger, abstract representations? Is it the same regions in the brain, or is it now new regions? Whatever it is, right? So that's kind of where we are in terms of looking at this. So I don't have data to share, but I thought it'd be interesting for you all to know that. So I will stop here, and I want to thank lots and lots of people over time. I've just been blessed with the most talented students, postdocs, undergrads, grad, PhD, all of that over time, and my very solid collaborators from Children's National, which is the children's hospital that is my clinical collaborator for like 25 years now. And then in this one study I told you, we collaborated with Stuart. Sorry, here we go again. And my funding, which who knows whether we will have any more of it. This is all NIH funding, the way things are in the US now. But anyway, just a big thank you to all my students. I'm just sharing a recent photo from the lab, but lots of people contribute to all of these studies, as you know. So thank you so much, and that's all I have to say. I don't know if you want to do questions.
Thanks,
Thank you.
Questions from the audience?
So you concluded with a relationship between generalizability and adaptability, right?
Can you hold the mic?
Yeah. So on your concluding remark, I just wanted to know, are you also in future going to work on the duration of this relation? Like if learning and adaptability are actually related, and
right.
for how long? Right?
Yes, yes. Yeah, so we are looking at that, right? So we do, so we have the third data point, like I said, but we are also following these kids for a fourth one, just looking at the adaptive functioning to see whether, whatever gains they make in adaptive functioning actually remain after intervention, because the intervention stops. So we will be able to link that. And I think the interesting thing for us is to see if anything in the brain actually predicts that, right? So that if there's something, let's say even at baseline, that actually predicts who improves more, so that's kind of what we're looking at, other than also just looking at where the changes happen. But we are really interested in that predictive question before.
Yeah, I noticed that very interesting talk.
Thank you.
Wide variety of methodologies as well as functions being investigated. One thing I see is that sensory motor network among the different resting state networks has the highest involvement, and as you said, these tasks have certain sensory motor goals, so it is interesting. What I see is the subcortical, SC is subcortical, has the least percentage. So, I mean, makes sense logically in the sense that the developing cortex has a lot more contribution. But is there some stabilization that has happened, and what is the implication of SC being only one?
No, that's a great question. And the SC there is primarily caudate and thalamus also. We didn't include the limbic ones because there was a lot of signal dropout around that. So just pointing that out. Yeah, I was actually quite surprised. And I think perhaps it just has to do with this general executive function state approach that we took, right? So that, you know, the caudate is important for, I mean, the shift, the stop signal, right? Like the motor inhibition per se. Not all of the other tasks need motor inhibition, right? So it just might have been weaker because we're taking this very general EF. It's an EF state, really, that we are capturing, right? So I think it may have something to do with that, and that's why perhaps the somatomotor may be so dominant. Yeah, we were so surprised, and then we went and looked in the literature quite a bit. It's interesting that, you know, somatomotor dysfunction is talked about in every disorder, but it doesn't come up as like the main thing because we tend to not think of it as sort of cognitive, right? But it's really important, and you may be aware of recent work from Evan Gordon, where they showed, and others, where they showed that the motor strip, right, the mapping that we thought, that whole homunculus, is actually not as straightforward. What they find is that within these effector-specific patches in primary motor cortex, somatosensory cortex, you have these patches of cortex that actually link to all of the major cognitive networks. Okay, so you have this interspersed mapping in primary motor cortex. It's not what we were thinking, right? So this is a find. I don't know if you guys are aware of this finding, right? Evan, by the way, was my PhD student, so I'm very proud and advertising but do look up the paper. I mean, it's beautiful work, but we have to change how we think about primary motor cortex and somatomotor cortex, right? So there's a lot of linkages to higher level functioning that are going on there, and perhaps that's why that, you know, we are capturing this. So we're still writing up, stuck at the discussion on this paper, because in terms of interpretation of these findings, you know, so one message of this thing is that it's very distributed, but I think there's some interest there. But I'm glad you were picking up on that. It's really, it was surprising, and it may just have to do with these tasks having to do with a lot of motor stuff, but.
Thanks so much, Chandan. Wonderful talk. So just picking up on the caudate as a comment, we haven't published that yet, but it's older work where we actually see the caudate show up in trilinguals in a stop task. You know?
Oh, okay.
And what language you should use in trilinguals. And the activity in the right caudate actually predicted their responses the best.
Interesting.
But I wanted to come back to a slide that you showed earlier about the subgroups. Okay?
Yeah.
And
you want to go back to that? Yeah.
Yeah, I saw that you sort of stayed away from saying anything about gender too much. But I was curious if in the 3 groups that you had, did you tend to see a larger number of, in the subgroups, more females in the flex emote versus the inhibit and metacog?
Yeah, we didn't. Okay, so do remember both. So the sample has ADHD, autism, and typical development. Both ADHD and autism are highly male prevalent, right? So the sample was predominantly male. The TD sample, we always try to match, right, to that, because we're picking up on studies. So we didn't see that. in this second study, the replication.
So the distribution across the three is roughly...
It's roughly equal, okay. Yeah. Yeah, it's roughly equal. I took that, it was a very busy table, so I took it out of the slide, but it's in the paper, but we didn't. Yeah. We didn't see that. In this study, the complex one, there's some differences that we are seeing, okay? I don't think it's heavily female, but there are some demographic type of differences. For example, it's, like I already said, it's more severe. Psychopathology, you could just do like an average of these domain scores or like area under the curve, and it's higher. It's also, they're a little bit older also. They also have more concurrent diagnosis. So in this, we're looking at that as well, how many comorbid diagnoses you have, which makes sense. I mean, you know, it's a more messier one. But I'm not seeing, we don't see a gender difference. And again, it just may be that the sample, I think, is more heavily male. Did I not put it here? Yeah.
Yes, you did.
Maybe,
actually.
yeah.
67%
So that's why.
I
But we
think
didn't
was male,
see that.
yeah.
We do have anxiety, depression, those as a primary diagnosis, but that's a small subgroup actually.
And just like a last comment, if in case, would you make a comment on how these subtypes kind of manifest in terms of the developmental trajectory?
Yeah, this is so interesting. I don't know. We haven't, again, I don't know, right? But that same Dunedin work, the Moffitt and others, Caspi's, that data set, what they've looked at is not, you know, obviously executive function related, but they've looked at psychopathology, right? Like what diagnosis you have at what year, what age, year. And what they find is that it's pretty dynamic. Like, you know, people come in and out of these symptoms. So you may have like some symptoms, say anxiety, depression peaking at age 13, but that same child at age 15 no longer meets criteria, and then they have something else, right? So it's a very fluid state, these symptoms, which kind of makes sense if you sort of think of them dimensionally and so on. So it'll be interesting to see the extent to which our ability to regulate, right, cognition, emotion, also kind of waxes and wanes a little bit. You would think so, right? Right. So yeah, I don't know, but I haven't. It's just such an expensive, time-consuming study to do, as you can imagine, to look at these longitudinally. So I haven't. Yeah. Good question, though.
Ma'am, I would first like to refer to an anecdote by Mr. Richard Feynman, in which he looked at his sense for count for sensing a minute, and he then entered into a competition with his friend, a mathematician friend, in which they competing whether they can accurately sense a minute, and then Feynman first, and there is some difference such that when Feynman was reading a passage, then he can accurately sense a minute, but his friend can't. But Feynman can do it while he was speaking. And his friend, which John Tukey, he can't sense a minute while he is visually reading, but he can sense a minute while he is speaking. So Feynman attributed the difference to the difference in their thinking style. Feynman said that he's more of an auditory type, and Turkey said he's more of an visual type. So I'm attributing it to the fact that in the functional adaptive model, there is the learning component and there is the abstraction component. So the abstraction component, I say, can it be related to that we all created different abstraction, different models in our mind to deal with the world. And some create more auditory, some create visual, and there can be other type of components that we use for the abstraction of the world. And if you look at the autistic population that are average and above average in IQ, so can it happen that the difference in their functioning, adaptive functioning, is due to because in the environment they are not sufficient models present that are in congruence with their internal internal abstraction model. So structural difference in brain, but the education does not provide them with the adequate models which can help them to improve their functional adaptation.
Yeah, interesting. You raise a really interesting point. In autism, certainly, right, there's strengths and weaknesses. And in terms of, whether you have sort of a good match with whatever teaching style and so on. So perhaps, I don't know what sort of the answer to that is in terms of what differences in the brain, but it's certainly conceivable that there might be some strengths that one particular child has, and the environment may not be suited, right, to that. And particularly in autism, we really try to pay attention to that and try to tailor the environment to enhance learning. So you raise a good point.
Thanks.
Thank you.
Thank you so much, Professor Vaidya. It's always a pleasure and listening to your work. Wonderful data, and we actually discuss your papers in class. Yeah, the data-driven one I remember particularly. Thank you, Professor Narayanan, for chairing this session. And we move on to our formal inaugural session now. It's my pleasure once again to welcome you all to this 12th Annual Conference of Cognitive Science and this inaugural session. I especially welcome Professor S.I. Rizvi. He is Dean Faculty of Science at University of Allahabad. May I request Dr. Shiv Sharma to formally welcome Professor Rizvi. We were also expecting Professor Manish Srivastav, Dean Research and Development, but he's not able to join, and also our Registrar, Professor Ashish Khare. So now we will move on to the next. Very pleasant to again welcome Professor Narayanan Srinivasan. He is President of the Association for Cognitive Science, and may I request Dr. Amrendra Singh to formally welcome Professor Narayanan. Yeah, we need this picture, Narayanan. Yeah. So I'll now invite Professor Narayanan to deliver a welcome address as the president of this association.
Thanks. I'll be brief. So this is the 12th conference. One nice thing is the association got started here at the center. So it's sort of nice to have the conference here at University of Allahabad. And I recognize two of my senior colleagues there who also played an important role for starting the center and developing cognitive science here. So this is just for the sake of students, not to embarrass you. Maybe you should stand up. Professor Janak Pandey, who is the first founder head of the center, is here. Oh, yes. And Professor R.C. Tripathi, who is also sitting next to him. Thanks. So great to see both of you. Okay, yeah, so we hope that the conferences will happen every year, and the numbers have grown every year. So it's good to see all of you, and I hope all of you have a very good time over the next 3 days. And yeah, thanks for coming.
Thank you, Professor Narayan. I would now like to invite Professor S.I. Rizvi to address the gathering.
Good morning, everybody. Welcome to this city of Prayagraj, earlier known as Allahabad. Welcome to the confluence of three rivers. And it is said that when you come to Allahabad, your cognitive faculties increase. I think this must have happened. You might be experiencing it. It is again a very happy moment for me to be speaking in front of two former vice-chancellors. I don't know, sir, whether you recognize me or not, but I do recognize Professor Janak Pandey, Professor R.C. Tripathi. Thank you, sir. When Bhumika asked me to say a few words, so by training I am a biologist, I am a biochemist, and I work in the area of aging and anti-aging interventions. So this is not my cup of tea to be speaking in a gathering of so many eminent people of cognitive science. But I do also write short stories and other non-fiction articles, and very often I use evolutionary biology as a cue. And when I was asked to say a few sentences about cognitive science, so I thought, what is cognitive science as a layman? And then it struck to me that it is something related to brain. And brain is one of those organs of which we know very little in biology. We have learned a lot of things about so many other aspects of human biology and other things we can understand, but we still have absolutely no clue that how these 86 billion neurons of this Homo sapiens means the wise human. And because of this wise human comes the term cognitive science. So cognition actually starts from the brain. And do all animals have this faculty of cognitive science, to understand what is cognition? So then I went back to maybe 7 million years back, that when a branch of evolution progressed and diverged from the existing chimpanzees, and we started becoming in the area of hominids. So something great happened in this era of evolution, that around 3 million years back, we became bipedal. And when we became bipedal, something great started to happen. We could walk, we could run larger distances, and then we started hunting animals. Before that, the diet of hominids was only vegetarian. We were not hunting animals. After we became bipedal, we started hunting animals and our diet changed. And because of the change in diet, large amount of energy, large amount of lipids became ingredient of our diets, and something great happened. Our brain started to grow. So the size of the brain of a monkey or a chimpanzee is around 300 to 400 grams still. But the size of the human brain is 1.3 kilograms. And as I said, we have 86 billion neurons, and each of these neurons, on a conservative state, they connect to 10,000 other neurons. So there's a huge amount of connections, and this huge amount of connections, what we actually developed is now what you all call consciousness. So we became conscious, but earlier animals are also conscious, but not in the sense that we are. And we started having different kind of emotions. And these emotions are part of your study, that how we express our feelings. So it is in this realm that I became interested in cognitive science. So something again great happened when we became bipedal. That was very important for the starting of our big brains. That we lost our body hair. So body hair is again important. If you go back, if you now try to imagine chimpanzees and gorillas and monkeys, you find that all of these animals have body hair, huge amount of body hair. But we started losing body hair. And when we lost this body hair, the dissipation of heat became much more easy. And when we could dissipate heat, so these animals cannot run for larger distances because their hair doesn't allow dissipation of heat. And because of this, so large amount of factors took place and our brain evolved. But then all those students, I would like to tell you that when you go back, for any kind of science, any kind of research, you must first try to go back in time and see the hugeness of the cosmic time scale. Because when we are talking about these things, we are talking of millions of years. And our mind is not that capable to understand what is 1 billion years. If I say something that something happened 1 year back, your mind doesn't take even a micro of a second to go back in time and realize what was. You can relate any, but since we have a finite lifespan, we just cannot go beyond 100 years. Then it becomes very difficult for you to understand. But 100 years in an evolutionary time scale is just a dot. So when we talk about millions of years, then we can understand how these things would have happened. And then some mutations took place in the brain. So there is an area known as Broca's region. And there is an area known as Wernicke's region. And there is a gene known as FOXP2. And the mutations in these areas and these genes, don't want to go into detail, but these things happened. And we found the ability to communicate. This was again a very, very big development in human history, that we started communicating. So it was just 10,000 years back that we started communicating like this. Just 50,000 years back, we were not communicating, we were not able to speak like this. And what we were doing, we were speaking in a way known as click languages. So we still follow these click languages, and suppose I am seeing somebody and somebody falls down. So from here, what I'll do, you will understand what I am trying to say. You will understand my emotions. But these are the click languages which were prevalent around 50,000 years back. And then, during this period, our larynx, for all animals, the larynx is very high in the alimentary tract. And our larynx descended to a lower place, and we started modulating our voice. So then, around 10,000 years back, these languages evolved. And so there is although you can see that there are so many different types of languages, they seem to be so different. But if you go to the beginning of these languages, you will find that the vowels came first, the consonants came later, and this is also apparent if you see the beginning of a child, when the child starts speaking. So within 6 months, a child born in any place in the world, speaking any language, starts speaking after 6 months vowels. And after 9 months, the child starts speaking consonants. So all these things have been the product of our evolved brains. And humans now are the only species on this earth who can think about the past, be in the present, and think about the future. And to encompass everything that I have said is probably why you are in this conference. All of this is encapsulated in something known as cognition. And that's why I have great respect for this Center of Cognitive Science. There have been several instances where you have diverged, you have changed the existing paradigm of this university. So, thank you so much, Professor Srinivasan also, for being instrumental in starting this society over here, University of Allahabad, and again, for bringing it back to University of Allahabad. So, thank you all of you. Hope you have a very, very pleasant stay in Allahabad. Thank you.
Thank you so much, Professor Rizvi, for your insightful words. Thanks a lot. I would now like to especially welcome Professor Manish Srivastava. He has arrived. He's Dean Research and Development in our university. May I request Dr. Niharika Singh to formally welcome Professor Srivastava. Also would like to mention we have some senior colleagues from the university, and most importantly Professor Janak Pandey, founder head of the center, Professor R.C. Tripathi, because it was the vision of these two seniors, distinguished scholars who actually thought about cognitive science in 2002, or actually 2000, not 2002, and the center came into being in 2002. I also recognize the presence of some of the heads of different departments. I can see Professor Shanti Sundaram, head biotechnology, Professor Neelam Yadav, head food technology, and Professor Neetu Mishra from family and community sciences, and Professor Manoj Kumar, he's IQAC director at the university. If I have missed any, Professor Chandranshu Sinha from Department of Psychology. So thank you for being here and for your presence here today. I would now request Dean Research and Development, Professor Manish Srivastava, to say a few words.
गुड मॉर्निंग टू ऑल ऑफ यू। सबसे पहले मेरे तरफ सामने जितने बैठे हुए रिसर्च प्रोफेसर लोग हैं उनको मेरे हृदय से नमन। ये 3 दिवस की जो कॉन्फ्रेंस में मैडम ने मुझे आमंत्रित किया है उनको मैं हृदय से शुक्रगुजार हूँ भूमिका मैडम का। सबसे पहले आज मैं केमिस्ट्री और मैथमेटिक्स का स्टूडेंट रहा हूँ, तो बायोलॉजी से मेरा कोई इंटरटच नहीं था कभी। बात होती है कॉग्निटिव साइंस की। कॉग्निटिव साइंस है क्या, उसके बारे में मैं कुछ कहना चाहूँगा। कॉग्निटिव साइंस एक तरह का मल्टीडिसिप्लिनरी कोर्सेस है जिसके अंतर्गत हम लोग कई चीज़ों का स्टडी करते हैं। फॉर एग्जांपल साइकोलॉजी है, फिलॉसफी है, कंप्यूटर साइंस है, आर्टिफिशियल इंटेलिजेंस है, अदर्स एक्सेट्रा। कॉग्निटिव साइंस में ये बताया जाता है कि मन, जो हमारा माइंड, मन को समझना पड़ता है। मन हम किस तरह की समझते हैं, हम कैसे किसी चीज़ को सोचते हैं, समझते हैं और किसी बात पे हम किसी निर्णय लेते हैं, हमारा मन कैसे काम करता है वो हम इसके अंतर्गत हम स्टडी करते हैं। स्पेशली कॉग्निटिव साइंस के अंतर्गत अपने मन के बारे में, ब्रेन के बारे में, ब्रेन क्या है और मन क्या है, दोनों के बारे में स्पेशली स्टडी की जाती है कॉग्निटिव साइंस के बारे में। इसके अंतर्गत हम ये कहते हैं हमारा मन कैसे काम करता है, कैसे सोचता है और हम किस तरह के लैंग्वेज में, किस तरह के भाषा का यूज़ कर रहे हैं कॉग्निटिव साइंस में। हम किसी से बात करने में, किसी भी ह्यूमन नेचर में, किसी भी मनुष्य के व्यवहार को समझने के लिए इन सब चीज़ों की आवश्यकता है। जब तक आप इन सब चीज़ों को नहीं जानेंगे तब किसी भी मानव के बिहेवियर को आप इजीली समझ नहीं पाएंगे। यह मानना है कि स्पेशली साइकोलॉजी जो होता है वो हमें स्पेशली आदमी के जो मस्तिष्क के बारे में नॉलेज करता है, उसके बिहेवियर के बारे में बताता है। पर जो न्यूरो साइंस है इसके अंदर जो न्यूरो साइंस का पार्ट होता है, न्यूरो साइंस हमारे मनुष्य के तंत्र के बारे में बताता है, उसके भी मस्तिष्क के स्ट्रक्चर्स के बारे में बताता है। और जो कंप्यूटर साइंस और आर्टिफिशियल इंटेलिजेंस का जो हम यूज़ करते हैं, जो स्पेशली आज का काफ़ी डिफरेंट टाइप के कंप्यूटर मॉडल्स बन रहे हैं। कंप्यूटर मॉडल्स की हेल्प से हम किसी भी ह्यूमन के बॉडी, मनुष्य के मस्तिष्क को, ब्रेन को स्टडी कर सकते हैं। इनके अलावा जो भी मशीन लर्निंग्स है, रोबोटिक्स है, इन सब का भी हम यूज़ करते हैं ह्यूमन नेचर में, आपके मन को, आपके ब्रेन को जानने के लिए। साइकोलॉजी के अंतर्गत मनुष्य के व्यवहार और मानसिक प्रक्रियाओं को समझते हैं, और न्यूरो साइंस के अंतर्गत हम मस्तिष्क के स्ट्रक्चर को जानते हैं। इनके अंतर्गत हम स्पेशली कई सारे इंस्ट्रूमेंट्स के बारे में हम जानते हैं, जैसे MRI है, EEG है, ये कैसे वर्क करते हैं, कैसे वो स्ट्रक्चर्स कैसे माइंड की स्टडी वो करते हैं। फिलॉसफी जो होता है बेटा, उसके अंदर स्पेशली हम स्टडी करते हैं कि आपकी चेतना क्या है, मन और शरीर के बीच में क्या रिलेशन है, कैसे हम मन को और शरीर के बीच में हम रिलेशन डेवलप करते हैं। तो इन सब चीज़ों के बारे में स्पेशली कॉग्निटिव साइंस में पढ़ाया जाता है, आप इसको जानते भी होंगे, समझते भी होंगे। तो मैं इनके साथ ये कहना चाहूँगा कि मैं इन 2 दिवसीय सेमिनार के सफलता के लिए सबसे अधिक दिल से हम शुभकामनाएं देते हैं कि अच्छे से संपन्न और आप कुछ सीख के जाए, जितने भी स्टूडेंट्स आए हैं वो कुछ अच्छा सीख के जाए, ये हमारी शुभकामनाएं आप सबसे। थैंक यू।
Thank you, Professor Srivastav. So before we end this session, we missed a very pleasant part of it. After the keynote, we would like to present a memento to Professor Chandan Vaidya. May I request Professor Narayanan to please. So this is a thing which students have actually made it. They have embroidered the brain, which I think you would appreciate. Yeah. It's Ananya who has done that. She is here. It's actually a cute little brain. Yeah, very nice. So we now move to the last part of this session. I request Dr. Amreen Singh to propose the vote of thanks, please.
Good morning to everyone present. Okay, so on behalf of Center of Behavioral and Cognitive Sciences, it's my pleasure to present this vote of thanks. So to begin with, I extend my heartfelt gratitude to our Honorable Vice Chancellor, Professor Sangeeta Srivastav, for her constant support and encouragement. I think her support has made this conference possible to organize in this hall. I'm deeply thankful to the President of ACCS, Professor Narayanan Srinivasan, for his constant support and encouragement, guidance throughout the process of organizing this conference. Sincere thanks to all our abstract reviewers. We received more than 210 abstracts this year, and all the abstracts went through a blind peer review by 2 reviewers. So that was a quite huge job, and thanks to all the reviewers for doing it on time, efficiently. We would like to thank Professor Chandan Vaidya, Professor Bharat Biswal, who came back within a month. He came for a Gyan course last month, went back to US and again came back. And Samar Hussain for taking out time and attending this conference and being our keynote speaker. Heartfelt thanks to our HOD for planning this conference, taking all the pain to, planning things and executing. So thank you, ma'am. Finally, I would like to acknowledge the support of our university administration, our registrar, Professor Ashish Khare, our finance officer, Professor Himanshu, all the deans present here, Dean Science Professor Rizvi sir, who takes time to always grace us, grace the occasions, whatever we organize, he is always there to bless us. Our Dean Science, Dean Research and Development, Professor Manish Srivastava sir, Coordinator of IQSC, Professor Manoj sir, all the heads present. Thank you all for taking out time and gracing this occasion. And finally, a special word of gratitude to our student volunteers. I think they are the architect of this conference. They have done all the hard work. All of what you are seeing has been done by our student volunteers. So big thank you to all of you for making this conference possible. So thank you all for being here. Thank you all for your presence and contributions. With this, I invite everyone to join us for high tea on the ground floor. Thank you so much.
Excuse me, please make sure that we are here back to the place by 11:30. We'll start the next session. Sorry, it's 11:20. Please be here by 11:20.
भैया नहीं ज़्यादा मत करो मगर इतना करो ऑनलाइन में दिखाना होगा आपकी दोनों
और प्रिंट आउट रखने के लिए जो
प्रिंट आउट आप इस पे रखिए आराम से टच है तो रुक जाओ बस नहीं पड़ेगी हैंड बैग चाहिए वो ले लीजिए अगर आप फिक्स होके ऐसे करें
हाँ वैसे तो
और ये ऐसे करेंगे तो ऐसे हो जाएगा ये देखिए इमेज हो रही ना
मतलब वैसे तो शीट यहाँ पे रख के शीट के हिसाब से प्लान है
जैसी रिक्वायरमेंट आप कर लीजिए दोनों मतलब सभी ऑप्शन अवेलेबल
हाँ
प्रेजेंटेशन अभी है आप
से बात हुई है। अबे तो वो वहाँ से चेंज करेंगे। लॉगिन कहाँ पे करना है?
कोई एक फिक्स रखो।
उसके लिए फिर प्रेजेंटर्स
किसी पे भी हो जाए।
नहीं, प्रेजेंटर्स तो वहाँ खड़े हो रहे हैं ना। ये क्या? ये वहाँ से है शायद, इधर से कंट्रोल
बुलाना पड़ेगा मेरे ख्याल से। ICT सेल। हाँ
मैं कर देता हूँ।
ओके, मिल गया। ठीक है हो गया। तृप्ति हो गया। क्या क्या करें? लिंक है या मैं भेज
मेल पे लॉगिन करना पड़ेगा।
लॉग देना। मैं संकेत तुम्हें भेजता हूँ। हाँ तुम भेजना, तुम ज्वाइन। और मैं आपको पहले इंट्रोड्यूस कर, ठीक है? हेलो, आई एम संकेत। आई एम हियर टू टॉक अबाउट माय रोल। एक पर रिलेटेड है ना, शेड्यूल्ड है ना ऑलरेडी। वो तो उनका है ना भाई, हम अपना करते हैं। ओह अच्छा।
और प्रेजेंटेशन मोड में भी जाना, फोन में कुछ करना होता है।
संकेत ईमेल
संकेत.p p या पवार संकेत.p। p और तुम भी
हेलो।
कौन तुम्हें वापस? तुम जितनी जल्दी कर लो। 11:20 तक वो यहाँ पे होना चाहिए।
इसका खत्म होगा, मतलब खत्म के क्वेश्चन आंसर सेशन स्टार्ट होगा, तब हमें मदरा को ज्वाइन करवा देना है।
ठीक है। एज इन कितने देर में? अभी से टाइमिंग बताइए।
टाइमिंग ये है, हमने उसको 12:20 गूगल मीट लिंक दिया हुआ है। ठीक है, तो 12:20 से पहले अगर ज्वाइन करवा
12:25 पे रिमाइंडर चला जाएगा।
मैं उसको कॉल, हाँ उसको वैसे जो मेल पे जाएगा रिमाइंडर वो अलग है, मैं उसको कॉल कर दूंगी कि बी रेडी, और फिर हम वो मेरे बोलने से पहले ज्वाइन करने की कोशिश नहीं करेगी।
ये क्या हुआ?
ये प्रेजेंटेशन मोड में है।
तो जैसे ही उसका क्वेश्चन एंड आंसर सेशन स्टार्ट होगा उसको ज्वाइन करवा देना, ठीक है? मतलब तो यही कराना।
हाँ
ठीक है।
संकेत, join the meeting.
मैं अभी उनके आने के बाद जाऊँगा फिर।
हाँ वो ठीक है, चाय के साथ कुछ खाने को।
लिंक मिल गया ना?
हेलो, हमारी फोटो आ रहे हैं हमारे।
What? तृप्ति, आप खाली हैं? मैं लिंक भेज रहा हूँ मीटिंग ज्वाइन करने का, वो ज्वाइन कर ले। जो उसका फ़ोन काम नहीं कर रहा।
ठीक है, भेजो। क्या कह रहे हो, समझ?
मैं वहाँ सामने से
सेम
ओके।
देख, कोई बात नहीं, हम नहीं लेंगे।
हाँ 10 मिनट रुको, मैं आता हूँ। ठीक, ओके। तृप्ति, हेलो तृप्ति, am I audible? क्लियर है ऑडियो?
हाँ, ऑडियो भी क्लियर है।
ठीक है,
मेरा ऑडियो क्लियर है?
हाँ आपका क्लियर है। एक बार प्रेजेंटेशन मोड पे जाइए।
एक सेकेंड, इसमें भी होता है ना व्हाट्सएप में?
माइक्रोफोन।
शेयर योर स्क्रीन।
Tripti, please introduce yourself and about your talk.
Hi everyone, this is Tripti. I will be presenting my talk on flexible modes of cognitive control. Is this okay? क्या काम नहीं कर रहा?
हेलो तृप्ति, अभी मैं ऑडिबल हूँ?
बोलो। Yes.
हेलो हेलो
हाँ, you're audible.
हेलो हेलो
You're audible.
हेलो
ठीक है, I think sorted.
हेलो
I can hear you.
ओके ओके हेलो हेलो अब आवाज़ आ रही है?
कनेक्शन
हाँ, you're audible. हाँ, आ रही है।
ठीक है ओके फाइन आ जाओ।
ओके, बंद कर रहा हूँ। ठीक है, कनेक्शन मैं हैंडल कर लूँगा, बाकी ये ईमेल्स का ये हैंडल करेगा। तुम इधर ही बैठना, मैं साथ में रहूँगा। ठीक है।
इसमें
हाँ
इसमें चला
नहीं उसमें
हाँ
ठीक है
और बस उनकी स्लाइड में दिक्कत थी, बाकी सब स्लाइड तुम खोल दो
देखने को अच्छा क्या लैपटॉप करना है कंप्यूटर करना है?
देखिए ये उसको छोड़ दीजिए IQC PPT ठीक देखिए प्ले कर रहे हैं
ठीक है बैक की इसको जरा एक
उसको देखिए ये इनकी कर रहे हैं
नहीं वो छोड़िए हमको अपने वाले में आने दीजिए
इनके में
ठीक है कोई बात नहीं हमको अपने वाले में आने दीजिए
कौन सी है
एक मिनट नहीं ये तो PDF है
अगर वर्ड है
ठीक है हाँ ठीक
तो भी इनका है
हाँ इन्हीं
नेक्स्ट
हाँ इन्हीं
इनवाइस कर दीजिए पहले कर दिया जाएगा
क्या
जब होगा तो कर देंगे
हाँ इसी में से खोलना है ना वो
हाँ वो सब चीज़ें कंप्लीट
हाँ ठीक है ओके अभी आप आ गए हैं यहाँ पे
हम हैं
मैं जा रहा हूँ चाय पे
जाइए
ये पेन ड्राइव किसकी है
नहीं पेन ड्राइव नहीं ये इसका रिसीवर है
अच्छा
वैसे अच्छा देखा जाए तो यही है
नहीं वो उन मैम को सिर्फ वो चाहिए था ठीक है हाँ मैं समझ रहा हूँ उन मैम को सिर्फ ये चाहिए था बाकी लोगों को यही चाहिए ठीक है
बाकी एक कॉलर माइक विकास भी रख दो यहाँ पे
वो है
हाँ नहीं यहाँ दीजिए जरूरत पड़े
सुनो
हेलो हेलो
ठीक है ना उधर भी ये फुल कर दीजिए ना
यार वो नहीं शो हो रही है अभी हटाना पड़ेगा अभी हटाना पड़ेगा हाँ इसको हटाओ नहीं इसी में चलेगी इसको हटा दो
हाँ कोई मतलब
नॉइल की है वो अपना आएगा चला लेगा है ना नॉइल का है अपना वो प्ले कर लेगा
Noel Tomi.
Yes.
पॉइंटर का यूज कर सकते हैं कीबोर्ड
पॉइंटर दे दीजिए
कीबोर्ड निकल के रख दिया यहाँ से भी कर सकते हो
ठीक है एक बार से करें
इससे भी हाँ और अगर इसमें प्रेजेंट करना मतलब दिखाना है कुछ इसे लॉन्ग प्रेस करके रखिएगा लाइट चलेगी और ऑन करना पड़ेगा इसको
ऑन एंड देन
ठीक है लॉन्ग प्रेस लाइट ऑन ओके ठीक है
So now we are going to start the first oral session on executive control, and I invite Professor Padmala. He's from Center of Neuroscience, IISc Bangalore. He'll chair the session on executive control. I'll hand over the mic.
Okay, welcome everyone. This is the first oral session on executive control.
This recording is going on, so if you stand here, you won't be recorded. Thank you.
So there is a small change in the schedule. The first scheduled talk by Ashwini from University of Hyderabad will be online. It will happen at the end of the session. So we'll start with the second scheduled talk by Noel Tomi from CBCS, University of Allahabad. So the talk is titled Quasi-Periodic Bold Patterns as Person-Specific Predictors of Sustained Attention Fluctuations. So each presenter will have 15 minutes of presentation time followed by a few questions, okay? So let's welcome Noel Tomi from CBCS.
Thank you, Professor. So here, I'm presenting my research titled Quasi-Periodic Bold Patterns as Person-Specific Predictors of Sustained Attention Fluctuations. So let's talk about sustained attention first. It's the ability to focus on a task consistently. But we know it's not like a spotlight. We have misunderstanding that it's like a spotlight, but it's more like a candle flicker. It wanes in and out. We have times of mind wandering, and we have times when we are in the zone. And these fluctuations are not random. they are mechanistic linear, they have a neural origin. But our current static functional connectivity measures, ways of measuring them, are not sufficient. This brings me to the measurement problem. Static functional connectivity, in a very basic sense, averages these phases. It sort of mushes together the whole signal and averages these sections of high attention and low attention parts together, resulting in a blurry picture, like you can see at the bottom. And on the other hand, dynamic functional connectivity takes samples the signal at discrete points, smaller points, and has a clearer picture. It's like a video camera compared to a camera. So is there a dynamic connectivity measure that is a better, that's in fact better to measure cognitive processes like attention, which are dynamic in nature? So here I would like to introduce quasi-periodic patterns. And these are discrete motifs or wave-like patterns in the Bose signal, specifically seen in the infraslow Bose signal, which is in the frequency of 0.1 to 0.001 hertz. These are 20-second long in humans, have been found in all mammals. It was found in 2011 by Mahid et al. And these are 20-second long in humans, 16-second in mice, etc. They are recurring. The time period between two patterns is random. That's why they're called quasi-periodic. These patterns normally involve a propagation of activity that switches between the default mode network and the task-positive network, or whatever constitutes task-positive network. And these are not noise. They're reproducible within individuals and observed across species. Because they drive the anticorrelation between internal and external attention networks, we hypothesize that they would be a superior marker for attention, probably, compared to static connectivity itself. For this study, the first question comes, how are we going to measure attention itself? So for this, we zeroed in on a task called visual metronome response task, in which a participant has to tap a button in tune to a metronome, a visual metronome, every 1.3 seconds. And this task provides us with a very dense, temporally dense behavioral metric. And the statistic, the behavioral metric that we calculate out of it is, from the reaction times, is reaction time variability. So every person could be fast or slow in their tapping, but what we're looking at is the stability of the tapping. So if they are stable, the reaction time variability will be low, and we assume that they are have higher levels of attention. And if the person has high variability, we say they have a relative attentional lapse in a particular session. I would also like to take a quick second to talk about the functional networks we're focusing on in this study. We're focusing on default mode network, the dorsal attention network, the somatosensory network, and the ventral attention network. Due to dearth of time, I won't go ahead explain what they are. Based on this, specifically the network pairs that we are working with are the DMN, all of these three networks, task-positive networks, correlated with the DMN itself. So we come to two main questions, the background questions of this study. First, do dynamic QPP metrics, specifically QPP metrics, predict attention better than static metrics? Not just as good as, but better than static metrics. And the second is whether they can act as an individual signature, a neural signature for each participant, because the relationship, every person's baseline is different, and so could be the relationship between the attention metric and the QPP metric for them. So first, a quick second to explain the task. Every participant had to tap a button every 1.3 seconds. A visual stimuli would be seen on the screen, which is a black box on a gray screen. And there were 10 blocks of 4.5 seconds each. And these blocks, each metric was calculated per block, so 10 data points per participant. For fMRI preprocessing, we selected a TR of 1 second that provides us with enough temporal resolution to resolve the QPP metrics, which are 20 seconds long. And for preprocessing, we had the typical preprocessing steps through fMRI prep, which are standard. But specifically, we did global signal regression and temporal bandpass filtering for the specific frequency that we require. And then each voxel had a time series produced. These time series were first parcellated using the Brainterm Atlas into 246 regions. And these 246 regions were then assigned to the functional networks based on EO's 7 hierarchical networks. And just a quick note on what the QPP extraction algorithm is. Simply, it's a template matching algorithm. It's not a sliding window algorithm, it's a template matching algorithm. Initial segment is a 20-second segment is taken, and it's slid across the whole signal. And the parts where the signal has a higher correlation to the template are selected and they're averaged together. This process is iteratively continued multiple times until we have a representative signal template, QPP template for the whole signal coming together. So for each data signal length, we have a 20-second long QPP template that's produced every time. And for statistical validation, we used linear mixed model, mixed effects model. And this was done because our data structure is nested. So each participant has 10 data points in themselves. And linear mixed models also allow us to test whether individual specific effects exist or not. So we have, specifically, we have fixed slopes and random slopes. Fixed slopes are about group level relationship. For example, we have beta value, beta QPP. So first, multiple models will be created. The first one will have only the static metrics as variables. The second model will have the QPP metrics as variables. And to that, we'll also add the random slopes as variables. And the performance will be compared to see whether there's an incremental improvement in performance with the addition of these variables. And the final test is a likelihood ratio test, because as we increase the number of terms in a model, the fit improves normally, but likelihood ratio test actually accounts for that. So coming to our first result, as we hypothesized, is an incremental value of QPPs over static connectivity. First, we did a simple correlational analysis, which is exploratory, and found that, the Pearson's R value, both the metrics show a relatively good high Pearson's R value, and the QPP metric does show a higher R value, more correlation with the behavioral metric. But the issue is the assumptions of correlation itself are violated in our data, in the kind of data set we have. So, therefore, we have to go with linear mixed models for this. And in it, we found that model 1, which only had the static connectivity variables in it, performed much worse than the QPP, the model with the QPP variable added to it. So we find that, and once the QPP variable is added, we find that the prediction, the ability of the static connectivity metric to explain variability is very diminished. So we can assume this from our analysis, that probably the variance or the correlation that is seen using the static metric itself actually encompasses, it borrows predictive ability from the QPP metric, which is QPPs themselves, which are in part of the signal itself. And when we resolve that, when we add QPPs themselves as a metric, we find that the predictive ability improves much more, because in a model which have variables which are collinear with themselves, a model which explains more variance absorbs the variance from the other one. So this sort of proves that QPPs are more predictive of the behavioral metric. For second hypothesis, whether each participant had individual relationship between the QPP metric and the person and the behavioral metric, we compared two models again, one with the fixed slopes for QPPs and static networks, and to that we added random slopes of person-specific for it to model person-specific relationships, and we found that again the second model performed much better. This proves that and as you can see, all the slopes are significantly different for each participant, which says that some people have a more resilient connection to QPPs, and some people have a more influential connection between QPPs and the metrics, etc. And as an extra exploratory edge analysis, just to see if we could find a mechanistic overview of why this happens, we did an edge analysis in which, so basically, we regressed the QPPs out of the whole network, and we then checked whether the contributional difference, any contributional difference occurs in the edges based on QPPs. So the contribution of QPPs in high performance, contribution of QPPs in low performance. 3,000 edges. We left it one edge surviving. Now, it doesn't mean that other edges did not have a significant effect, but with that high of a number of comparisons, Benjamin Hochberg has a tendency to become too restrictive. But for this study, because it was mechanistically interpretable, we focused on this for now. So the connection was between the putamen, dorsolateral putamen, and the S1 trunk area, the primary somatosensory trunk area. And the putamen, as we know, is implicated in timing or rhythm maintenance. So it also aggregates information from the motor planning area, supplementary motor area, from the somatosensory area, and then from the input it receives from somatosensory area, it guides the basal ganglia to set a specific rhythm. And this is the connection we found had a huge effect size, a coincidence of 2.4 is huge, in fact, and that's probably why it also survived the edge analysis. And we hypothesize, coming to it, based on this, we have a view that sort of, we can look at QPPs like a global weather in the brain. Global weather in the brain. So depending on the weather, depending on the anticorrelational state, how anticorrelated they are, they sort of modulate local connectivity. So at a global level, you have the weather, and it modulates local connectivity, which translates into or influences behavioral performance itself. And that's what we found, and this is how we interpret it. We can have questions about it. And again, the implications about this are whether this could be a new interesting biomarker for attention itself. And these have been used to classify Alzheimer's and classify patients with and without ADHD already in 2018. But this could become a more consistent so right now, we'll come to it in a bit. So first I'll talk about limitations. We had a small sample size of n is equal to 13. So the issue is the result that we found that the slopes were varied. We need a higher sample size to confirm that. So replication is needed with a higher sample size. And all the results that we found are probably task specific. The edge that survived is task specific probably because it was a visual metronome task. But the QPPs are not task specific. They are a general characteristic of the brain itself, of the cytoarchitecture itself. So the study should be done with different cognitive tasks. And I talked about the strict FDR correction part. So again, coming to future directions, we have to see whether this applies to different tasks, whether these QPPs in an individual level can be used to measure the level of diagnosis or pathogenicity, etc. One second. Yeah, so the biggest implication, the idea with which I started this research was that I wanted to see if there was a non-behavioral way to measure attention. Right now, all of our methods of measuring attention are behavioral, or we use a static connectivity-based metrics to measure trait-level attention, trait-level, sort of like IQ. We measure trait-level differences. But can we see where, relatively, when the person was in their task, just from the fMRI data itself, when the person was focused on a task, and when the person was relatively out of focus in the task? And won't that actually be helpful in our further studies? So yeah, this is, yeah, thank you.
Hi, thank you for the presentation. I wanted to know whether you are hinting at intra-individual variability in QPP processing or inter-individual variability. Then I'll ask the other question.
Intra-individual.
Intra-individual. Yeah. So do you also feel that this prediction is also variable with time of the day?
Oh, definitely. So there is a search that functional connectivity boundaries itself change over the course of the day, etc. So the baselines and the relationship could change over the day itself for each participant. So variability will be involved. And now our challenge as neuroscientists or cognitive scientists is to model specific methods or understand these relationships and then model them better so that we can predict them better in the future. But yes, to answer your question, these will show change.
So with account to your study and variability with time of the day as well, right? And of course, assuming that, there's also intra-individual, then inter-individual, and then time of the day, so many variabilities. What is the implication, overall implication?
So I would like to give an example of, let's say we become very good at using QPPs the way it's shown here. Again, I don't know if it's likely, further research needs to be done. But let's say we have a memory task, or let's say we have a cognitive task on model decision making in the fMRI scanner. Now, for a lot of tasks, the levels of attention, modulating or maintaining the levels of attention are pretty important. And attention becomes a confound that we never we just have to sacrifice it, right? So what if there was a method to understand where in the scan the person was more attentive and where in scan the person was less attentive? And that can be used as a covariable of sorts, maybe. So that is the overall idea, long-term idea that I started this research with.
That's quite interesting, because then you are hinting at studies of different kinds can also have continuous, instead of time-locked and stimulus-locked analysis, we can also have continuous observation, and that can be used as a covariate. Thank you.
In the study, I specifically selected these blocks because the QPP template still does a sort of averaging. But we can possibly, if we push the algorithm a little further, maybe decrease the length of the blocks itself, or maybe isolate individual events or instances of the pattern and use them use them specifically. So yeah, maybe we can improve the temporal resolution itself, but this needs further research.
Sorry. Very nice work. So I'm wondering, you have 50 minutes of data?
50 minutes of data.
Which is a lot, right?
Yeah,
so, the limitations of getting good data for that long.
Yeah.
Do you have some sense of how much, what is the lowest amount of data that you can actually see a link with the
So before this specific data collection study, all the studies on QPPs have been on a group level, where they concatenate small sections of data of multiple participants and find a QPP on a group level. So this is the first study I know which has tried to do that on an individual basis or within an individual. So that is a question. We have been, in my data.
You can look at it. You have the data.
Yeah, in my data at least, stable QPPs have been. I selected 4.5. I did not try going below it. 4. So every 4.5 minute scan has given me stable QPP data.
Oh, just one 4.5?
So every QPP metric, every QPP template is generated for each block. So for a 5-minute block, this study was able to generate a stable QPP.
Okay.
Yeah.
I'm happy to hear that, but I'm a little suspect. I will talk more with you.
Okay, we'll move to the next talk. Let's thank Noel one more time. So the next talk will be by Adraja Chhaterji from IIT Kanpur. The talk titled Disentangling the Ego versus Allocentric Navigation Switching Performance and Relating to Cortical Energy Resource in Mild Cognitively Impaired Patients.
This one?
Okay, let's welcome Adraja.
Hello everyone, good morning. I am Adraja from Translational Neuroscience and Technology Lab, Department of Cognitive Science, IIT Kanpur. And my topic of presentation is distangling the ego versus allocentric navigation switching performance and relating it to cortical energy resource in mild cognitively impaired patients. So, So I'll be firstly giving a background on, it's fine. I'll be firstly giving a background on MCI population. So MCI is the transitional state between normal aging and dementia characterized by cognitive decline, which is like they're not as like greater than normal aging but not meeting the criteria for dementia. The stage is marked by subtle deficits in memory and spatial navigation or decision making and other cognitive functions. So to be able to carry out these functions efficiently depends on the ability to manage the cognitive load. So and we see that this ability to manage the cognitive load decreases, sorry for the interruption, my whole slide is not visible, can you just... But it's fine, my time will go. It's okay. So this ability gets increasingly compromised in MCI, this ability to manage their load. So that causes a strain which further accelerates cognitive decline. So this ability to manage the load depends on a person's available cognitive resources. And a major challenge is to relocate these resources and the competition that is there between the relevant and irrelevant stimuli for these resources. So, inhibitory mechanisms normally suppress such interference, but this inhibition declines with aging. And with MCI patients often struggle to suppress this irrelevant stimuli and thoughts, reducing resources available for task-relevant processing and thereby accelerating cognitive decline. So our aim is to overload them in such a way that challenges their resources and study how these resources are distributed under load. So the overarching goal was to identify potential behavioral and neural markers under load that distinguish the MCI population. So our core task was a spatial navigation task. We chose spatial navigation for two reasons. One, because it is a multimodal process and involves a lot of cognitive functions like decision making, memory, mental rotation, and also spatial navigation deficits are one of the early symptoms in MCI. So to sufficiently overload them such that their resources are challenged or to cognitively choke them, we had an additional working memory task, which is the digit numbering task, and also we had a background passive auditory mismatch task that measures error-related potentials driven by unexpected auditory cues. So I will like to talk about this in a bit more detail. So this was a tone that, this task, MMN task that we played in background did not require active engagement from the participant. It was like a proxy measure for available cognitive resources. So it was a tone that comprised of standard tones and deviant tones. Deviant tones were larger, rare, and randomly placed. And it was like a surprise element. So the brain expects the standard tone, and so the more stable the memory trace for the standard tone is, the stronger is the expectation, and larger is the mismatch when violated by deviant tone. And when we get the ERP difference between the deviant and the standard tone, we see that it's more negative when the violation is greater. And so we had this as a measure of the proxy measure of the available cognitive resources. So I'll be also giving a bit of introduction about the terms that I'm going to use, which is important for like navigation. So navigation relies on two distinct components, egocentric and allocentric. Egocentric is based on self-centric measure. So with respect to the individual, like you can see this right side picture, where is the cylinder with respect to the individual? But the allocentric is a world-centric view. Like, where is the cylinder with respect to the maybe spare? So, and also what is important is the flexibility to switch between these two frames while we are navigating. So I'll just give a real world example to give you all the intuition of flexibility in switching. Is so if I'm going to a cafe in a mall, so I'm thinking, okay, I'll take a left and then I'll go here. So and then suddenly my friend calls and she says that, okay, sorry, yeah. So it's across the bookstore and north of the fountain. So now I'm thinking based on the map representation. And when I cut the call and again start moving, I come back to my egocentric viewpoint. So our main hypothesis was how cognitive load affects this flexibility to switch between egocentric and allocentric frames, and how the cognitive resource availability measured via MMN using EEG relates to this switching in MCI. So yeah, moving on to the methods. We had participants greater than age of 55, 14 male and 10 female, and we divided them into MCI and healthy based on MMSE questionnaire, mini mental state examination. So if they scored below 23, they were considered MCI, and if they scored above 23, they were considered healthy. And we did some baseline test for our working memory load profile to check their upper and lower thresholds, right? How many digits a person can remember with 100% accuracy was our lower threshold, and how many digits a person can remember with, cannot remember, 0% accuracy was our upper threshold. And we also did a reference frame proclivity test, which was to check their intrinsic preference preferred reference frame, which is egocentric or allocentric. So we had 5 trials that had working memory loads provided in the order standard, which was same for all. High load, low, and high. This was personalized per participant according to their baseline working memory load profile. Also, the number of turns kept increasing by 3 per trial. So we are calling this as navigational load in the study. So in total, there was 5 trials with this 2x2 design, and the MMN audio was played throughout the task. So before every trial, the participant is asked to be mindful of 2 landmarks that are on the eye level. So we probed them about the 2 landmarks. And during the trial, they were narrated the sequence digit by digit to avoid chunking, so that it creates the continuous involvement of the load that we want. And after the trial, we asked them to recall the sequence we recited. And there were 2 questions on navigation, that is egocentric measure and allocentric measures. So egocentric measure is where is the lift, just with respect to the individual, and allocentric measure is where is the lift if you are standing in front of the fire extinguisher you encountered on the way. And as I said, before every trial, we probed them on 2 landmarks. So here the fire extinguisher is already 2 landmarks. Okay, I'll be making it quicker. So we had these measures, and how we calculated egocentric, the flexibility to switch was, if they performed better in the non-intrinsic reference frame, we called it a switch. And then we also got the ERP measure by epoching our event, standard and deviant tones, and between like 100 and 400 milliseconds. And the measure we used was the difference between deviant and standard. So coming on to the results, MCI was associated with reduced likelihood of switching, which we expected because switching is a more costly phenomenon, and we expected them to switch lesser than the healthy population, and it matched the observed distribution. This model was done on, trained on 80% and tested on 20% of the data. We also saw interaction between the load and group. So under high load, MCI participants switched more, which we found surprising, and we thought this as of a breakdown of their stable strategy. And to investigate it further, we saw that ERP amplitudes, we investigated the neural levels. We saw ERP amplitudes were more negative in MCI, and so that means they were allocating more of their resources in the MMN task rather than the switching task. And here the MMN was the irrelevant stimuli that we wanted them to load with, like wanted them to challenge their resources. So to get a better idea of the spatial distribution, we performed a generalized eigen decomposition, and we saw a parietal dominance relative to frontal activation in MCI under load. So if you see the low MCI, the highest bar that is there, they have parietal activity which is very dominant, and during switch, they were switching less during low load, and which we think as an efficient spatial strategy. But during high load, this parietal decreases and we see a switch. More interestingly, what we found is like frontal activation became more pronounced relative to parietal. So, relative to parietal, so it was positively correlated with the navigational pattern. We think may reflect a compensatory shift with areas taking over when they're insufficient. Summarizing my study, although navigation impairments are hallmark of MCI, the of intrinsic space and to switch between has been explored less. Our study is conducted in a real-world setting, making it more ecologically valid. MCI patients showed less flexibility to switch from their intrinsic frame than healthy cohort in general. So we expected this, and we didn't think that they'll switch more, which is a more costly process. But this behavior reversed under high load, high working memory load conditions, which we found as an interesting result because it suggested a breakdown of their stable strategy. We can further validate this by our GED and MMN findings, where we showed that parietal dominance decreases during high and are being allocated to the MMN task rather than the switching task or the spatial navigation task. In addition, frontal activity gets more pronounced to parietal. Shift account for the insufficient resources in parietal. Together, these results highlight impaired resource allocation, disrupted frame switching, and reliance towards frontal from parietal due to insufficient resources as early markers of cognitive vulnerability in MCI. And our future studies will include that some connectivity analysis and coupling analysis between frontal and parietal activity and relate it with this flexibility of switching to get a better idea of the activities. Thank you. Questions, please.
Hello, Adraja.
Hi.
Good talk. So, first question I had is, did you take their baseline of egocentric and allocentric abilities? Because I did not follow that in the...
Yeah, so not egocentric and allocentric abilities. Maybe I missed it because of time. But we took a basic spatial awareness test, that is direction of sense of direction test. Yeah, to understand their abilities. Yeah.
Why did you choose MMSE and not MOCA? Because in MOCA they would have been part of a clock test also, which would have helped you to identify in general their abilities. Was there any particular reason?
No, actually.
Okay, and like when you frame the question, right, where is the lift? And you are calling it an egocentric question. Did you have any metric from where this has come? Because from my understanding of spatial navigation, when you ask me where is the lift, there can be an ego as well as allocentric, both abilities I can use to think of and come up with the answer. So how do you determine the specificity for this?
Okay, so we can't, because we had old participants and we need to make them understand. So we had a practice trial where we specifically mentioned how they should think of the two questions. Okay, so we said, okay, where is this chair with respect to you? And then we also had other objects in the room and said, where is this chair with respect to that thing? So we kind of gave them a practice so that they understand it better, what we are asking. Yeah.
And with aging, from what my understanding is, there is more egocentricity, right, as you age. So you have taken the MCI, which is 55-year-old and above on an average.
Yes.
Did you have healthy matched controls?
What? Sorry?
Did you have healthy matched controls to...
Yeah. We had 13 MCI and 11 healthy participants.
What were the average age of the 11?
Average age of
11 participants that you taken? They are also older population?
Yeah, so there was not significant difference between groups. But
in age?
In age, yes.
Okay. Yeah.
Thank you.
Thank you.
Hi, hello. Here. First, I need a clarification. You said switching, right? So I didn't understand. What do you mean by switching? What they were switching? Can you clarify?
Yeah, so navigator, like everyone has a intrinsic frame that they prefer, like allocentric or egocentric, and the switching is preferring the non-intrinsic frame with respect to the intrinsic one. So if they switch from the intrinsic, that we call as switching.
Okay. So in under high load condition, you found that the switching was more under high load condition?
Yes. Okay. In MCI.
In MCI, right?
Yes.
So, and you said that it's a surprising finding for you, right? I mean, were you able to interpret?
Why will they, when we load them more, and why will they take to us a more, a costly process of switching under high load.
Yeah.
But then when we saw the neural correlates and how the thing were coming up, we could understand that this as a breakdown of their stable strategy, because if it was a stable strategy, they would have not switched, which we saw in general.
So actually, I could interpret your result. Maybe I can help you. So if you know this perceptual load theory of selective attention by Professor Neely Levy, so there is two kinds of, one is perceptual load and another is the cognitive load, which you have manipulated, which is like you're loading the memory, right?
Yes.
So according to this theory, when you load it more, then you lose the control and then you are more distracted.
Yeah,
right? Then maybe this could be one. And MCI, anyways, they might have less attentional resources anyways. And you are loading even more, then they must be more distracted, right? Then maybe this could be one of the mechanisms that why it is happening under high load.
Yeah, so we also saw that, if I can move again, sorry. So we saw that it was related with more navigational errors, and therefore we thought that it was a breakdown of their strategy, and I agree with your point, what you're saying. Yeah.
Let's thank the speaker. We'll move to the next talk.
Thank you so much.
Which file? इसका भी PDF आपका PDF है कौन सा है अमोरा टावर PPT ऑन करिए देखिए
So the next speaker will be Amora Tawar from University of Hyderabad. The talk is titled Partners L2 Proficiency Modulates Executive Control in a Joint Task. Let's welcome Amora.
Good morning. My name is Amora and I am a PhD student at the University of Hyderabad, and today I'll be presenting my work which is titled Partner Second Language Proficiency Modulates Executive Control in a Joint Task. Yeah. So I want to begin by talking about what joint action actually means. So any form of joint action refers to any form of social interaction between two individuals when they're coordinating their actions to bring about a change in the environment. And because they're performing these actions in the same space and time, they end up sharing a lot of representations with each other. So it's not just that they're sitting together performing a task next to each other, but it's also that they're sharing a lot of task goals, their intentions and their actions as well. And these shared representations are referred to as co-representations. Essentially, joint action is a powerful tool to study shared cognition. Yeah. So I'll talk a little bit about previous work that has been done in joint action. So in a seminal study done by Sebans et al. In 2003, they actually performed a joint Simon task, and it was found that when two individuals were performing the same task together, a spatial compatibility effect was observed, and the same compatibility effect disappears when a single individual performs the same task. Similarly, a joint flanker task was performed in 2011, and it was found that it was not just their flanker stimulus was interference in their task performance, but also their partner's flanker stimulus was also causing interference, even if it doesn't have anything to do with their task goal. And in a NAVON task, it was found that when your co-actor's focus of attention is different from your own, your performance is disrupted. So essentially, your partner matters in whatever task that you're doing together. And if your task goals are not aligned, then you might end up disrupting performance. But this large body of research has not taken into account a very socially relevant cue, which is language proficiency. So talking about proficiency, we know that bilinguals constantly manage interference from two different languages at any given time. And this constant interference has resulted their control mechanisms to be very adaptive in nature. So depending on the context and depending on the communicative context and the social context, they tend to exercise a more sort of adaptive control. So it has also been found that second language proficiency is modulates executive control. And bilinguals use this sort of executive control to maintain their task goals more effectively. So in this current study, what I aim to do is to bring together the adaptive control view of bilingualism and the co-representation view of joint action. And I asked if linguistic asymmetry changes how people implement executive control. So the paradigm that I chose for this study is a joint flanker task. So as we all know, in a classic flanker task paradigm, flanker task essentially measures monitoring and inhibitory control. So in a joint task, two participants are sitting next to each other, sharing the same screen, and they're performing the task simultaneously. So I ask how performance will be affected if co-representation and executive control are put in direct contact with each other. So my experiment had Telugu-English bilinguals from the University of Hyderabad. The population was entirely university students. And the experiment was divided into two phases. Phase one was when we collected their language measures. So they came in and we collected their Lextail data, their verbal fluency, semantic fluency, and we asked them to fill out the language social background questionnaire. And all of these measures together, we calculated a composite score based on which they were classified as either low or high proficient, high L2 proficient speakers. And they came in for the second phase of the experiment, which was the main task, the joint flanker task portion of it. And participants were divided into three categories, three groups. So essentially, high and high paired together, high and low proficient paired together, and low and low paired together. And before the main task, they were asked to interact with each other for 5 to 10 minutes so that they could judge each other's language proficiency. So this was my design. I had a fixation cross followed by the flanker stimulus and then a blank screen. The flanker stimulus was given in two colors, red and blue. So for example, participant A was given red to respond to and participant B was given blue to respond to. And they were instructed to respond to only their colored arrows and ignore the partner's color. And trials were randomized. So my independent variables here are the proficiency of the participant, the flanker trial type, which is either incongruent or congruent trials, and the switch type of trial. So we define here as a switch trial and a stay trial is that if a participant's trial is preceded by their own trial, it's a stay trial. And if a participant trial is preceded by their partner's trial, then it becomes a switch trial. So essentially what we're trying to see if how performance differs when they're moving from the same context from one trial to another, and how performance differs when they're moving from one trial in a particular context to a different context. And we measure reaction time over here as a dependent variable. And repeated measures ANOVA was performed with post hoc pairwise comparisons. For the results we found, as we can see in the first graph, a flanker effect across all groups and proficiencies. So essentially the task was doing what it was supposed to do. People were exercising their control mechanisms, and so we can see that congruent trials have faster responses than incongruent trials. And similarly, when we analyze the switch and the stay trials, surprisingly what we found was that participants were faster in switch trials when compared to stay trials, and this was a common finding. But it was not uniformly seen across proficiency, as I will talk about later. Yeah, so essentially what this means is that when they're switching to a different context, somehow it's facilitating their performance. Yeah, so in the high and the low proficient conditions, we see that there's not much of a difference between their stay and their switch trials, which essentially means that the moment you're a symmetric pair, you're not showing that much of a difference in your performance across these stay and switch trials. But when we had a significant interaction between proficiency and the switch condition, so what we saw surprisingly was that low proficient participants seemed to be more flexible with their control mechanisms. So low L2 proficient participants were faster in switch conditions significantly than the stay condition, and this was not seen in the high L2 proficient participants, which means that somehow high proficient participants were more stable with their control mechanisms, and low proficient participants were actually more flexible, and they might have made use of some additional control mechanisms to sort of deal with the asymmetry in the situation. Yeah, so our study shows that executive control is not necessarily a fixed individual phenomena. It's more contextual, it's more interpersonal, and much more socially adaptive. And participants adapted their control processes depending on their partner's proficiency. This was especially seen in low L2 proficient partners. Symmetric pairs did not show any modulation. And it seems like if there is any sort of social asymmetry in a particular context or in a situation, that has triggered the use of additional control mechanisms and encourage participants to be more flexible when compared to a social context where more or less linguistically everything is symmetrical. So yeah, this is my study, and I would like to thank my supervisor and my lab members for all their support. Yeah, I'm open to any questions.
Yeah, nice presentation. Thank you. Were the participants required to communicate during the task?
No, they were not asked to communicate.
The second thing is, do you have a baseline non-joint setting measurements from these participants as well?
So I personally have not included that in my task because such tasks have already been done in the wide...
No, but not in this setting, right?
Yeah.
In the bio.
Yeah, but our main focus was to compare the kind of pairing that was done. So we didn't have the aim to compare a pair versus an individualistic view. So we tried to compare the symmetric pairs versus the asymmetric pairs. So that was the focus of our attempt.
The last question is, how do you think the bilingual status flows into this actions that you perform in this kind of flanker task? What is the implication that you see the differences?
Sorry?
The differences that you observe depending on the pairing has, what is the bilingual status, proficiency status, how does that flow into the actual execution of the task? You should...
Yeah, well, I mean, it's clear that language plays a very important role acting as a social cue, even if we don't realize it as much. So I think based on your partner's proficiency, based on who you're talking to, you somehow subconsciously adapt to their proficiency and their style of talking and their tone and everything, and basically you navigate your social context according to that. So I think that itself becomes like an important factor to consider in like such a setting.
Hi, nice presentation. Just following up from Professor Bapiraju's question. So I was wondering, why did you not choose a linguistic task?
Well, we wanted to sort of try to connect these two different bodies of work, which is executive control and the adaptive control thing. Maybe my next experiment could be something
Because
like that.
as sir said, language proficiency, there's not a direct bridge between this task and how language proficiency factors
Yes,
in.
yes.
And language proficiency is not overtly visible, so I'm not sure what kind of adaptation you're hinting at. So maybe you can look into it.
Yeah, definitely.
One final question.
Hello. Hi. This is interesting study presented. Just out of curiosity, I was wondering about the nature of the task that you selected. So you mentioned a joint task, right? But by the sounds of it, the performance and the task that was done was sounding pretty individualistic, right? So wouldn't have some sort of a collaborative task have been more efficient in this sort of a study?
Yeah, definitely. We were looking into that. But the point being that I was talking about co-representations, right? So when you are performing a task together, having the same goal to finish the task, whatever instructions that were given to you, you end up sharing these representations with the other person who is seated next to you, even if you're not necessarily communicating during the course of the experiment. So that is what we wanted to, sort of test out, how it would play, how all of this interplays with each other.
So don't you think that this is also dependent a lot upon personality factors rather than linguistic factors per se? Or I mean, was there some sort of a match, like, pretty much like some sort of a match based upon which the participants were paired, and then so that we can factor out only language elements from their performance?
Yeah, I mean, it could be personality based as well, but we tried our best to keep everything consistent and just change the language proficiency. So it remains to be seen. I mean, if you take each of these personality factors as variables, then you can sort of understand how they play into, something like this.
Thank you.
Okay. So let's thank the speaker, Amora, again. So the final talk will be online presentation by Ashwini Madira from University of Hyderabad.
नहीं आप करिए
ऑफ करना है नहीं
अपना लाया ना उधर हो गया उस पे उसमें भी ओके या सर यहाँ पे काम कर रहा है कैमरा एक ही जगह करी तभी तो होगा नहीं बंद कर दो क्या इसको चेंज करो इधर इधर यूज कर लिए हैं ये लोग
Hey Ashwini, can you share the slides?
Yes, sir. Is this visible?
Yes, ma'am.
Can we get
Slide
full
is visible.
Can we get full screen? That's okay, that's okay. We are good now. That's fine. Okay, so this will be the last talk for the session. Talk is titled Reactive, Not Proactive Suppression, Eye Tracking Evidence for Attentional Control Strategy During Visual Search by Ashwini from University of Hyderabad. You can go.
Okay. Hello, everyone. I'm Ashwini, PhD student with Professor Mishra from University of Hyderabad. Firstly, I would like to thank the organizers and ma'am for enabling us to present online. I'm having a lot of echo coming from there. I can hear my own voice. Hello? I can still hear my voice very distinctly.
Yes, ma'am. Any problem?
There's an echo coming from your end.
1 minute, ma'am.
Yeah.
Ma'am, now it is okay.
I think Yeah, I would like to thank the organizers and ma'am for enabling us to present online. Unfortunately, I'm not able to be there in person. And today I'm going to be presenting our research investigating proactive versus reactive control strategies adopted during distractor suppression. So, to begin the presentation, I would like to discuss what exactly is suppression. Our visual world, as we know, is filled with bright colors, sudden movements, and unexpected shapes, which are always competing for our attention, and yet we seem to be protected from the chaos. So, amongst the information that is thrown at us, we anticipate some of it, and we also are. There is some information that is unexpected to us. We do not expect it. So, for instance, I am seated in the seminar hall, let's say, and I would expect to hear a person talk, and that is information that I am expecting. Whereas there would be some noise coming from the audience, which I'm also expecting, but I would likely suppress it. So, here, what is happening is these distractors, whether unexpected or unexpected, are blocked for better processing of the speaker's voice. This mechanism is what is referred to as suppression here. And we also observe this kind of suppression when we are at the supermarket searching for groceries, or when we are at a parking lot searching for our car. Now, why exactly does suppression happen? Why do we suppress information? So, owing to the limited processing capacity of our system, information competes for attentional resources such that we engage goal relevant information and protect the resources from getting occupied by the goal irrelevant information. So, this is purely with the idea of processing the target more efficiently. So, the information here creates a competition, what is relevant versus what is irrelevant. And based on our goal, we tend to select that which is relevant over the others. Precisely what happens in both auditory as well as visual mechanisms. Now, there are different models of suppression that have been discussed so far. One is your signal suppression hypothesis, which talks about how visual system can proactively inhibit the salient signals when there is an unexpected or an expected distractor, preventing it from capturing our attention so that we are able to better focus on the target. Similarly, information that is matching the attentional template that we have created, that is also getting more weightage compared to the information that does not match our attentional set. This is what contingent capture theory says. And we also have priority map models where these different information, whether we have the top-down goals or bottom-up salient features of the object, all of these come together to create a certain weightage for the target over the distractors. And what exactly the whole study is this modified the signal suppression hypothesis that Kasplin and colleagues had recently proposed. The main aim primarily here is that does suppression actually mean that we have some information we are attending to and rapidly disengaging from it immediately? Also, another question that this particular model raises is that when we attribute certain attentional weightage to the information, is it primarily guided by the target, or is it guided both by the target as well as the distractor? So, is the target guided allocation of attention and the distractor guided allocation of attention happening parallelly? Another question here is that how do these mechanisms exactly manifest? What is the underlying process through which this kind of a suppression is happening? So there were different paradigms that were used to measure signal suppression, and I will be discussing them as well in some time. So this new version, the signal suppression 2.0, shows that the control of spatial attention occurs via modulations of feature signals, both bottom up and top down, and location signals that occur via different learning mechanisms. So, what exactly is the kind of control that is getting engaged over here? The main idea here is that we have Braver's dual control model. We have proactive control as well as reactive control. And which of these mechanisms is getting manifested or engaged when we are trying to suppress distractors? Is it through a top-down suppression of sensory representations that we are a priori doing? That is, even before the stimuli, let's say, comes in the visual field as a perceptual stimuli, are we trying to suppress it? Or is it as a reactive mechanism that suppression happens only after I see something salient and not before? So the main idea here is whether we are doing a bottom-up versus a top suppression. These theories culminate together to create the motivation for our study. And we are trying to examine the temporal dynamics of attentional suppression and trying to understand the difference between proactive versus reactive suppression mechanisms. Paradigms, predominant paradigms, commonality of these paradigms is that they have used both either feature search or a singleton, where you're looking for an odd shape paradigms to understand whether the suppression is happening anticipatory, that is proactively, or reactively. However, they do have a mechanism to distinguish the suppression occurring before the search array even appeared on the screen versus suppression that is occurring after the stimulus has been presented on the screen. So, like you can see, you have the Hung et al. Paper, for instance, which looks at the singleton paradigm, additional singleton paradigm, and they have presented probes and cues. Though the cue is being used to identify the potential location where the stimulus is likely to come, the distractor suppression as such is only being measured after the search display has occurred on the screen. Similarly, for other paradigms. What have we done new? So we have created 2 experiments where we are trying to separate this mechanism of anticipatory and proactive and reactive suppression strategies. We have used 2 experiments. The first is where I can again hear my own voice. Yeah, thank you. So the first is where under 100% predictive cues, we have tried to measure anticipatory proactive suppression. And in the second experiment, we have induced some uncertainty by modifying the cue validity. And we again measured for anticipatory or reactive distractor suppression. So we hypothesized that if the distractor suppression operates proactively, that is before the screen has the stimulus on it, the onset of the stimulus, then observers will direct anticipatory gaze away from the predictor distractor location prior to the search onset. And this will also be biased, the anticipatory period will be biased away from the distractor's expected position relative to other non-salient distractors. Similarly, if the distractor suppression is reactive, then the anticipatory gaze will not show any kind of bias between salient and non-salient distractors, and it will purely be guided by target-based attention. So this is our design. I'll take you through this. So initially, the experiment started with a fixation displayed for 500 milliseconds, and we showed the preview of the potential search array for 500 milliseconds. The participants then saw a cue which asked them to rotate mentally this particular search array. This was the primary and most important criteria over here, which enabled us to work on differentiating between proactive and reactive suppression on the representation of the stimulus rather than the visual display of the stimulus directly on the screen. So when we asked the participants to rotate, they were given a cue. They were asked to rotate the search array by the different degrees that you can see over here, 0, 90, 180, and 270. And they were pseudo-blocked for the clockwise as well as anti-clockwise directions. And once the cue appeared, a blank screen for 1000 seconds where they did the mental rotation, and then the search array appeared for 2000 milliseconds with the rotated search array. And the rotated search array now had orientation of lines inside each shape. they were told that you have to search for the square, the target shape, and press A if the word. So this, as you can see, created three different scope points or focus points for us to understand the fixation movements of the participant. One is during the preview, where they just had the glimpse of what the target shape is, where it is located, and where the is. And during the blank, we wanted to look at their potential eye movements towards the anticipatory location of the target, and they could also anticipate the potential location of the salient distractor here. Now, coming to the findings. So the details, we had 30 participants and we created 80 trials, each in 6 blocks. That gave us around 480 trials. And we had 20% of the trials where the salient distractor was absent, so that we know that they are engaging in the main mechanism of suppression initially. The results, so this is during the final search array, where we first analyzed the reaction times to see if they engaged in suppression or not. We did find that they did show significantly faster performance on trials where the salient distractor was present compared to the trials where the salient distractor was absent. Once we got this assurance that yes, they did engage in suppression, we then traced back to see where, how, and through which strategy did this suppression occur. Yeah, so this is the eye movement fixation data. We did a time course analysis on the proportion of fixations towards the target averaged across the non-salient distractors and the salient distractor. And as you can see, in the first preview phase, this is during the preview phase over here, as you can see, we did see capture of attention. There is eye movements towards the target, but when you compare their fixation patterns towards the non-salient and salient distractors, you are seeing a significant proportion of eye movements towards the salient distractor. However, when we go to the blank screen, they are showing no signs of suppression, and they are completely focused, or they are guided completely by the target's potential location. So they are not differentiating between the salient distractor's location where it might come or the non-salient distractor's locations. In the search screen, we saw minute signs of suppression of the salient distractor, and there is direct eye movements towards the target. So this was the initial findings from the first experiment. And
yeah. Ashwini, you have just 2 more minutes. Hello, Ashwini, can you hear me?
Yes, sir.
Yeah, you have 2 more left in the presentation time. You should rush.
Okay. Okay. So the main findings here is that we did not see any evidence of proactive distractor suppression, and there was attentional capture by the salient distractor. Now, in order to check whether this particular phenomenon could be because the participants had 100% idea, so it would have been a better strategy to just rely on the target rather than to rely on the distractor. So we induced Q uncertainty. The experiment design remains the same with minute changes, which is we add 5% congruent trials where the Q could indicate whether the search array rotated in the exact same direction as the Q's indication or not. And here we found that performance was faster on the congruent trials compared to the incongruent trials. And this was the case in both distractor present as well as distractor absent conditions. This is during the search screen on the reaction time. Having established this, we analyzed the same fixation patterns using the time course. Interestingly, we found that the distractor suppression during the preview had a cross interaction where initially we saw significant suppression and then the suppression reduced. So this suppression was primarily mapped to the Saccard planning phase between the 50 to 200 milliseconds window. And in the blank screen, we did not see any difference between the salient distractor as well as the non-salient distractors. And in the search screen, we saw significant suppression of the distractors again. This was what changed between the first experiment to the second experiment. Now, the main findings here are that behavioral evidence again supports distractor suppression, yes, but reactive suppression emerges at the search display onset. And we did see some sign of suppression, but only during the preview phase, which did not extend to any anticipatory proactive suppression. And this kind of gives an information that proactive suppression depends on the perceptual visibility of the cue, and we are not really guided by the knowledge alone, unless and until the salience here is featural, that is, we are able to see it perceptually. The idea of suppression is not running parallel. So the theoretical contributions, it does clarify the temporal nature of distractor suppression. It refines the conditions under which we have proactive suppression. So it emerges only, like I said, when there is and just pure knowledge about a distractor being there might not really play a strong role to elicit suppression. And also only when the task is, let's say, uncertain or difficult, this elicits the reason to suppress the distractors. Otherwise, we are purely engaging just the target, we are focused only on the target, and we are guided by the target's location and features alone. So yes, this offers a boundary condition for priority map suppression theories, and that's us. Thank you. I'm open for questions.
Anyone has quick question? So I just have one question.
Yes, sir.
I'm just curious because typically even the dual control model suggests that engaging proactive control is metabolically costly. Suppose in your first task, if you had offered some motivational incentive, right, would you expect that participants could potentially engage in proactive suppression?
So here we are, I think, adding, if I'm understanding the question correctly, we are adding another variable of reward unless and until there is an implicit motivation for the participant to engage in proactive suppression. And if it proves economical, only then I would say the reward would help. Because like we have seen, even when there is a no reward condition like we have done, in the second experiment, because there was an uncertainty factor, when they were looking at the preview screen and the blank screen, they did not know whether the next trial would be congruent or incongruent. In which case, they had to rely on the strategies, extra, let's say, resources in this case, to identify and make sure that they do not get captured by the salient distractor and focus only on the target. So in this case, even if we add a reward, it might elicit a stronger proactive suppression in the second case, but I don't see why it should elicit a stronger anticipatory suppression in the first case, the first experiment.
Okay. Yeah, looks like there are no more questions. Let's thank Ashwini one more time, okay? So this concludes the first.
Thank you.
Yeah, this concludes the first oral session. Let's thank all the speakers one more time for one interesting set of talks.
I would like to thank Professor Padmala for chairing the session and guiding the discussion so smoothly. And a warm thank you to all the presenters of this session. And now we would break for lunch and we would expect everyone to be back by 1:45. Thank you. Just a small announcement more. All the poster presenters who are presenting today, please put up your posters before the start of the next session. Thank you.
नहीं नहीं ठीक है.
ये तो बड़ा मस्त है. ये VIT में होता था हमारे उधर, लाइब्रेरी में भी था ऐसे एक टेबल.
हम लोग हैरान हो गए थे ये देख के.
VIT में था ऐसे.
Please move to the lunch area. So, ma'am, क्या? एक मिनट अनाउंसमेंट करना है. ये लोग
अनाउंस करना चाहती हैं.
कर रहा हूँ.
आइए मैम यहाँ पे हम लोग अनाउंस करना.
Everybody, can we please move for lunch? The people who are still here, can we please move for lunch? Thank you. नाम ले सकते हैं? नाम ले सकते हैं? नोएल लंच.
कौन रह गया? नोएल खाना नहीं है क्या?
बस हो गए ना, अब तो हो गए.
हेलो हेलो.
थैंक यू. चल ये बंद हो जाएगा. ठीक है.
इसमें ये इंटरेक्टिव है नीचे वाला, मतलब यू कैन, तो बेसिकली ये पूरा सिस्टम है तुम्हारा, यू कैन कंट्रोल।
हाँ ये तो ये है, ओ नाइस। और ये क्या है?
ये
भाई कौन है?
तो
हेलो।
ये इंटरेक्टिव नहीं है।
अच्छा ये टच नहीं है, ये वैसे ही देखने के लिए होगा ना?
हाँ मतलब अगर किसी कोई दिक्कत ना हो इसलिए ऐसे लगा दो।
हेलो, हाय। कहाँ से बंद होता है ये? ऑन ही रहने दूँ? क्या ट्रिगर्ड, कृपया ध्यान दीजिए।
को बोलिए कि मीटिंग लिंक अभी तुरंत भेजे. बाहर ही पड़ा है. ACCS email. हाँ ठीक है. मीटिंग लिंक सेंट आइटम में होना चाहिए. हम लोग पहले से लगा वो सर आएंगे तुरंत चालू.
सर तो आएंगे पहले इन दो का करेंगे.
नहीं मैं कह रहा हूँ इनको मीटिंग पे लिंक करके छोड़ के हम छोड़ दें. तुरंत जैसे वो ज्वाइन करेंगे हम ज्वाइन करेंगे स्टार्ट.
अब बताओ शैलेन.
अभी वही कर.
बंद करो माइक ऑफ करो.
बंद कर.
हो गया इससे भी आएगा.
Madhavi ma'am, can you hear me?
Yeah, I can hear you, Tripti, yes.
Okay,
Yeah.
so everything is fine for this call. So we'll start your session at around 2:40, right? Yeah, yours on the
Yeah,
is on the third number.
Okay. Yeah, it's on the third number, yes.
Yes,
yeah,
ma'am.
240.
So
yeah.
I'll just ping you once we are about to start. Okay?
You can hear me well, right?
Yes, yes. We can hear you and we can see you as well.
Okay, yeah, because I'm logged into the Ashoka email ID also because just that to just see the audience actually. Otherwise, I can actually connect from there also. Yeah.
So you're able to see the
Is this
audience?
better?
You're able to
Yeah.
see the audience,
I
right?
can see the audience. Yeah, I can see the audience.
Okay,
But
okay.
when I share the screen, do you want me to share and show once?
Ha, you can try once.
Yeah, because I don't want anything to. There is this one. Can
Yeah,
you
we
see
are
them?
able to see your screen.
Yeah, you're able to see the screen,
Yes.
right? Okay, perfect.
Okay, so for now, we are stopping the session and we'll get back to you on the third number, right?
Yes, yes. So I'll join by 2:30.
Yes, okay.
So in case if there is anything, then I can immediately join
Yes, okay,
by
ma'am.
2:30.
Okay, thank you.
Thank you, Tripti. See you.
Okay. क्या करने बोले? अभी कोई आया नहीं सर कौन सा पोडियम यूज़ कर रहे हैं वो ही है ऑनलाइन ऑफलाइन यही कर लें चेंज कर दें ठीक है लैपटॉप ले लीजिए मैं उधर सेट कर रहा अभी किसका टॉक है एक शीट मेरे को दीजिए और वही उसी वाले पे करना है
Then you should ask. नहीं आप पूछो फिर ना मैं कैसे No. Is Gori Gungjal here? Hi, okay. And then this is
This is Madhavi Lata.
Madhavi Lata,
She will be joining
online.
online. So we have checked.
and Prajapin. ठीक है, people are.
Welcome back, everyone. So we move into our next oral session now, which is on language processing. I invite Dr. Samar Hussain, Professor, IIT Delhi, to chair this session. Thank you.
All right, good afternoon, everyone. Welcome to the post-lunch session. We'll have 4 presentations in this session. So I will request the first speaker, Palab Basu, who is going to be talking about syntactic expectations in Bengali, probing filler gap dependencies using BERT. Please, Palab, go ahead. Palab, you have 15 minutes, right? And then followed by 5 minutes of Q&A. Yeah, 15 minutes, 15 plus 5. Okay.
Hello. Yeah. Just want to present it. Present mode. It's all right. Yeah. Good afternoon, everyone. Today I'm going to present on syntactic expectations in Bengali, and I am going to look at filler gap dependencies using bidirectional encoding models. So this is the roadmap. We will first talk about the background, and we will spend some time on the background itself. Then I will go to the methods. Then I will discuss the results, and at the end I will conclude. So just let us look at this verb, devour. So whenever there is a verb which is transitive, which requires 2 things, one an agent and one a patient, it requires a devourer and a devouree. And these arrows are basically dependency arrows. So a verb is a head which has 2 dependents. One is the agent, the devourer, and one is the patient, devouree. So let's look at this sentence. I know that the lion devoured the gazelle yesterday. And it's easy to see that the gazelle is the object of the verb devoured, and the lion is the subject or the agent who devoured the gazelle. But stark difference comes when you have a word like what instead of that. I know what the lion devoured. Now you cannot have a word after devoured like the gazelle or anything, the hyena yesterday, because that would make the sentence ungrammatical. So what is happening whenever there is a word like what, I know what the lion devoured, the word is actually acting like a filler, plus filler, and then whenever you have a plus filler, your workspace starts searching for a gap, and then you have a gap just right after the verb. And motivating on that, these dependencies are mostly always like formed and influenced by multiple type of processing constraints and cognitive constraints. And most important, these 2 constraints are most important. One is the linear locality and one is the hierarchical structure. So let us go back to this, and you see there is a linearity between the devour and the gap. It's just linear. So linear locality is driven by a quick resolution of dependency relationships between the head and the dependents, between the verb and the dependents, and the filler and the gap. And this is known as active dependency formation. On the other hand, sometimes these dependencies are also resolved based on some hierarchical constraints. So let us look what is hierarchy. So then this sentence, I know what the lion devoured yesterday, what comes before the lion and devour. But actually what happens, I know what has an answer to the what was the patient of the event, what was devoured by the lion. So it should come after the verb devour. So inherently we can, instantly we can see that there is a movement, subtle movement. The what is somewhere should come right after the verb, but it is on the surface structure it came way before. So this is the hierarchical constraint on processing. So let us look at this sentence now. My brother wanted to know who Ruth would bring. Now, you immediately will try to integrate who with the object position of bring. Who Ruth would bring? Ruth is bringing someone. Then no, you see us. Home to at Christmas. So whenever you see us, it gives you a surprise. No, you are surprised at the us. But if you consider the sentence B, instead of who, when you have if, there is no surprise. So what happens whenever you have a WH element, when you have a WH element which questions a noun, with the answer to the question is a noun, and if you don't see the gap instantly in your search space, it gives you a processing difficulty. So in a self-paced reading study, one would expect that us would give a higher reaction time with respect to a baseline condition B, because it is deeply embedded in a prepositional phrase right after to. So without this kind of complex structures in English, this linear and structural locality converges. So the first example I started with, lion and the devour one, in that example the structural gap and the local gap are the same thing. So I motivate on this, and Bengali is probably the best test case where hierarchical and linear gaps actually do not converge always. So let us look at the language background. It's basically a head final language where subject, object, verb is the canonical structure, but it has a flexible word order. You can scramble things. Embedded clauses can appear pre-verbally and post-verbally. And what does that mean? Actually, you have two markers. So in Hindi, when you have a sentence like Palab ne socha ki wo aaj ACCS mein present karega, that ki is basically the complementizer, and that ki gets translated to bole and J in Bengali. Bole comes in clause final and J comes in clause initial. So let's look at this interesting thing also, that is case marking. So you have case markers and any patient gets a K case mark. Similarly in Hindi, ko case mark, Pallav ne Naina ko kitab diya. Ko is always attached to the patient, the receiver of the book or any event. And phi, the null is no case mark. So whenever you have a case mark with a WH element, it looks for a gap. It searches for a gap. And without any case mark, there is no search. So basically, the clauses look like this. Pallav thought that Pranav gave Naina the book. And you see the J comes right after the main verb think. But the same sentence can be put like this. So you can see a mirror image kind of a thing where actually the main verb goes at the right end. It's right extrapose kind of a thing that you wait all the way for the end of the sentence to come to see the main verb. But the embedded clause is introduced way before the main verb. So even you don't know what Pallav did, but you don't know what actually happened. And there are two specific markers, J and Bole, and you can see the case marker K attached with the receiver of the book. So now, these are two kinds of gaps in Bengali that I actually experimented in this work. Is like when you have K marked WH Kake, it looks for a gap. Now, gaps can be linearly available, gaps can be structurally available. So, these two sentences are ambiguous sentences where you can have two gaps. The black line indicates the structurally available gap, linearly available gap, and the red line indicates the structurally available gap. And you see the first sentence with J, which is very similar to Hindi ki, which is pre-verbal. There, the search space gives you an option to have the linear gap in a linear order and the structural gap far away from the verb. But when you have the reverse order with the bole, the linear gap actually becomes anti-local and the structural gap becomes local. So you can have a prediction that given these two conditions, if you manipulate the WH word with a WH word which does not look for a gap, just like kaha, kab, when and where, it should not look for an element, right, in your search space. Then this, with this compared to this baseline, this would give you certain kind of surprisal value when you actually don't see a gap. And if you see a gap, then you are good to go with the language. So based on this, I used Chacon et al.'s framework, and basically there are two kind of gaps and two kind of clauses. one matrix verb comes first, in one clause embedded verb comes first. And I manipulated the clauses like when the argument is k marked WH, like kisko, kake, versus argument which is non-case marked, like when and where. In linguistics we call it adjunct phrases. Basically they don't require a gap, so they don't start a search operation in your space. And the embedded verb position, matrix first, and minus matrix means the embedded first. Basically it's a 2x2 design, and I had 25 items for each 25 items for each condition, so 100 constructed stimuli. So this is what it looks like. So I employed mask modeling task where I put the mask where I am expecting a gap, and I asked the BERT model to give me 5 top predictions. Why I asked it 5 top prediction? BERT looks at both directions and give me a particular word, and I actually look for a word which is non-case marked with K. Because as I already have WHK in condition A, it should have something which is non-WH or non-K marked. Because anything which is non-K marked is good for me, and I give it +1 point. If I get something like NPK, some NP with a K mark, any kind of NP, animate, inanimate, human, non-human kind of NP, it is -1. So it's bad. And similarly in condition C, when argument is there, the matrix first is there, this is something going to be a problem. So this is the task, and I got 5 top predictions, and I aggregated the probability based on this filtration criteria. And this is the second experiment where I actually masked the right end of the sentence. The very right end of the sentence I masked them to get a surprisal value, because BERT is a model that it looks throughout the sentence. So I made the BERT act like a autoregressive model that would give me a probability value based on the context, and here the context is only the leftmost context starting from the WH word till the gap position, and I took only those continuations which are suitable for my sentences. And this yielded a structure sensitive estimate for model's expectation of the gap. So the prediction is encountering a K marked WH phrase will make the model anticipate and gap, and the higher probability for argument like prediction as the mask side, and lower surprisal as the mask position due to fulfilled expectation from the experiment 2. So I used linear mixed effects models, and I used some contrast for the analysis. And this I am really going to the visualization first. And you see for the matrix condition, the argument probability is very less in comparison to the non-argument probability. So whenever you see a K marked WH before, you look for a gap and you don't get an argument with K marked anymore. So argument probability goes down, and as probability goes down, as you are taking surprisal, which is negative logarithm of probability, surprisal goes up. So lesser probability and with higher surprisal. So that's why the higher surprisal means that whenever you saw a higher surprisal, that means the model didn't actually do well. So now you can see there is also a effect of clause order. For embedded first condition, it should be like matrix first. Its embedded verb also doesn't play any different role in my language, but the model probably is expecting a huge number of K marked arguments in comparison to the matrix first condition. Model 2 performed well, but not as per the expectation, and it only yielded the argument condition, not the clause order effect. Model 3 performed very bad, and it predicted something else. So all 3 models, they differ. And model 1 shows the strongest structural sensitivity both in hierarchical and linear, but in a subtle way it failed to capture the structural expectation, because the embedded first condition actually yielded a lot more K marked argument elements that I am not expecting there. It should be something non-K marked. Model 2 partial sensitive, capturing only the linearity effects and some structural effects, but not the clause order and the interaction of these 2 effects. And model 3 shows minimal evidence for any kind of expectation or argument driven prediction. So the key take is this model 1 distinguishes gaps and shows clear clause order modulation that a human being will also show. It exhibits a meaningful interaction between argument type and clause order. And experiment 2 model also shows a robust clause order effect on gap surprisal. These are the model specification. So the Electra style model, because it's a self-supervised way of tokenization, probably that's why this model is performing well than custom tokenization. And I don't have access to all these data sets they're trained on, so can't really talk about what exactly is inside the model. But more or less, these models are tested on some benchmarks which are developed by computational people, not exactly linguistic benchmarks but they should be tested on, what recent literatures are showing, at least in English. So probably these models suffer for this kind of thing. And secondly, the embedded verb condition that with the Bole complementizer is something people don't consider it as standard when they are writing something. So there is a clear-cut difference also between the dialogue and text. So when you produce something in speech, in real life, you go through a different route, probably when you take a different route in text. So that is also clearly shown. So yeah, I really want to thank Pranav, Naina for their support, and Ishani, Ishita, all my batchmates. Yeah, lab mates, sorry. Yeah, thank you.
All right, thank you. Palab, we have time for some questions.
Hi, Palab. Thank you so much. In your evaluation, right, you have this W conditioned on the context. And I think you showed that the context is from the WH word down. I don't understand Bengali, so I was trying to follow on as much as I could. What information is getting lost by leaving out the parts before? Do you have any, for example, an object or a subject or something that comes before? So what is information that you're losing out by taking the context only from WH onwards?
Yeah. So basically, bidirectional encoding representation models actually look at the both side of the mask. So if I have a sentence like, I dash the book the whole night, the most expected filler for that dash would be, I read the book whole night. But if you have a word like, I looked at the book for the whole night, that gives you a surprise. No, who looks at a book at the whole night? So you expect read to be there. So when the whole night or anything right after the mask should not influence my behavior of the model, that's why I mask the right part of the model. Because as I am not using an auto-recursive model like GPT models, they use transitional probability from one word to another word. So the probability at the ith word is dependent from 1th word to t minus 1th word. But what it will do, it will also take from t plus 1 to the nth word. And that might give you a good prediction. But for human being, if I think a human being is listening to a speech or reading a text, they go through this thing in a piecemeal fashion. So they take one word at one chunk at a time. So that is why I mask the right end. Yeah.
Time for, yes.
Yeah, sure.
The one after this, where you gave the first example about us. Yeah.
Yeah.
So this is where I got stuck, and as a consequence in the middle, I lost out.
Yeah.
I would have thought the natural sentence structure is, my brother wanted to know who Ruth would bring home to us at Christmas.
Yeah. So this
Is that right in my assumption?
So why this is happening to you, and this is very normal to happen. You are expecting who to be resolved as soon as possible.
Okay.
But this sentence is grammatical in English. My bad, I did not add the citation. This is from a study. Stowe showed that when you have this kind of a sentence, so in linguistics we call it island extraction. You have an island, you take one thing from one island and you cross the river and put it somewhere else. So it's very complex to think. So what will happen in a study like when you have one word per key press. So you have a big sentence, you key press and words come and they disappear. So you will see my brother, then you press the key, you see wanted, then you see to know, then you see who. Now whenever you have seen who, your search space got initiated. You have a probe and you now look for something where you will mitigate this problem. You want the problem as soon as to be mitigated. Then you see Ruth. So you see, okay, this is a noun, let us see the verb. You saw the verb would bring. Now you want to mitigate the problem. And whenever you see us, this gives you a surprisal. In predictive processing, you call it surprisal. In behavioral domain, you call it high reaction time. So basically, the signature that you are getting the continuation is actually right continuation. That is the most expected continuation.
Okay.
Yeah.
And for my own interest, I'm interested in trying to see if, as a Bengali speaker,
Yeah.
you then went on to learn English.
Yeah.
Okay. How might this impact your sentence structures?
So as
in this context, in terms of surprise?
Yeah, so I want to go back to this thing. So I have these two sentences. So in Bengali, we can have us, like for this Pallav thought that Pranav gave Naina the book, you can have two constructions. And both constructions are equally likely good they don't need any context. So what used to happen to me, I remember, in my language, at least in spoken domain, I always drop J because it's allowed. Pallav thought Pranav gave the book to Naina. But when I was in school, I remember I used to drop that. But in my school, teachers were not linguists. They didn't think that can be dropped. But in English, that can be dropped. Actually, in spoken discourse, that can be dropped. But in written discourse, you cannot. Secondly, what happens when you form questions? So we have this kind of questions in English. What did Pallav assume that Pranav right after the conference gave to Naina? So you have this what way in the left side and you have right on a gap somewhere else. You are standing in Allahabad and the gap is in Delhi. You can't do that. In English allows that. But in Bengali, you don't do it. You put things in a linear order. For psycholinguistic experiments, you manipulate things in a way to see the interaction of the memory or the prediction, how it works. But in real life, probably as a Bengali, I would never say a sentence like, who Pallav gave book. Then no, I just put the WH right at the gap position. Probably that's the linearity that we go for.
Thank you very much.
All right, thank you, Pallav, for your talk. We'll now request the second speaker, Gori Gungjal, who is going to be talking about processing of emotional expressions in Hindi L1 and English L2 bilinguals.
You can use this, you can use the वो जब ब्लैंक हुआ तब क्या
उसमें आपने किया है उसमें है अपलोड है
फुल स्क्रीन कर दे ओके ठीक है Good afternoon everyone. I'm Gori Gungjal from Multilingualism and Literacy Lab, IIT Gandhinagar. And today I'll be presenting our work titled Processing of Emotional Expressions in Hindi L1 and English L2 Bilinguals. So just to give you all a brief context, research in bilingualism has consistently shown us that a first language is more emotional than a second language, and it is also like much more preferred for inner speech, for recalling memories, and for emotional expression as well. But then we also have research suggesting that the emotional strength of a language can be, it can match the level of this first language in certain contexts, such as when it's used in close relationships and
and in a much more frequent manner. Yeah. So, and then we have skin conductance response studies which show that L2 can evoke strong emotional responses, particularly in proficient speakers. So let's take a closer look at what defines a meaningful context. There was this study by Ozanska Ponikvaya where she demonstrated that a second language can evoke strong emotional responses. Her study was particularly focused at immigrant populations and individuals who were immersed in different cultural environments. So coming to the Indian multilingual context, especially in the Hindi-English bilingual context, we see that Hindi is typically used in personal and familial contexts, while English is more used in the professional and academic context. So we wanted to build on this natural contextual divide and examine whether emotional expressions, emotional reactivity is, if it's shifting based on the language as well as the context. This leads us to our main research question, which is, does emotional reactivity to Hindi L1 and English L2 change depending on whether the context is personal or professional? So we hypothesized that depending on the context, we'll see differences in emotional reactivity across languages. So our participants were 12 native Hindi speakers, and they all had native level proficiency in Hindi and were educated in English medium schools from K to 12. And they all reported high proficiency in English. For measuring emotional arousal, we used skin conductance response, and we also asked our participants to fill a modified version of a LEAP Q. For the stimuli, we used materials derived from real-life interactions in personal and professional contexts. And a study was modeled after Harris's 2004 experiment, wherein they had introduced 4 categories of emotional expression, specifically taboo words, reprimands, endearments, and insults in the Spanish and English context. In addition to those categories, we introduced the contextual distinctions of professional and personal, and we retained those categories used in Harris's study under the personal context, and we identified acclamation, disapproval, and confrontation under the professional category. So a significant portion of English expressions were taken from Harris's study, while the Hindi expressions were chosen based on their daily usage and relevance in a day-to-day life. All the stimuli were selected to elicit emotional arousal by stimulating realistic interpersonal and professional interactions. And then moving on to the experimental design, we followed a 2x2x3 factorial design where language, context, and expression types were the 3 factors. And we also included control words about emotion in both the languages. And here we can see the type of expressions that we had used. Yeah, so the experiment began with a fixation cross, which stayed on the screen for 1 second, and then the stimulus appeared, followed by a blank screen. So that made up one trial. So so the specific timings were chosen to match the typical dynamics of the skin conductance response. Yeah, the experiment was divided into 5 blocks of 30 stimuli each to prevent habituation and carryover effects. And then the participants were seated comfortably at an arm's length, and they were instructed to read the expressions as if they were being said to them in a direct conversation. And the activity was recorded using EDA electrodes, disposable EDA electrodes, that were attached to the fingers of the participants. And the electrodes were connected using a transmitter which was attached to the wrist of the participants, which was in turn attached to the hardware. And the software was also configured for acquisition. Yeah, and then we were giving, so the software, the ACCS12-2025 software was configured to detect the event markers that we were going to, that would be corresponding to the stimulus that we were showing the participants, and that was done using MATLAB. So once the data collection was done, we went through these data pre-processing steps where we had to code and convert the binary codes into the decimal categories, and then we did some baseline correction to extract the phasic skin conductance response. Next, we normalized the data using z-score transformation. And then finally, we averaged out the SCRs within each language by context condition. And our key findings were that we found a strong interaction between language and context, as we can see here. We found that participants had strong SCRs to Hindi stimuli across both the contexts. But when it came to English stimuli, it elicited comparable SCRs only under the professional category. In personal context, English stimuli produced weaker SCRs compared to Hindi. We also looked at the effect size across both the categories and the languages, and we found that English professional elicited strong differences in emotional reactivity. So the SCRs were higher for English professional than English personal, and then for Hindi personal and Hindi professional, there was almost no difference between SCRs. And also, they were higher, the SCR was higher for English professional than Hindi professional. And finally, SCR was higher for Hindi personal than English personal as well. So the effect size was the largest for English personal versus English professional, and then English personal versus Hindi personal, and then English professional versus Hindi professional. And lastly, Hindi personal versus Hindi professional. We also looked at the control conditions, effect size of the control conditions, where we found that the control conditions were eliciting weaker responses than the main stimuli. And then we also looked at the correlations with proficiency and age of acquisition, where we saw that most SCRs were not strongly predicting, were not being predicted by proficiency or age of acquisition. But there was an exception that a higher English proficiency was indicating, like it was pointing towards strong SCRs. So if somebody was if somebody was much more proficient in English, in that case, the SCRs were in personal context, personal English context, the SCRs were coming out to be higher. We also saw some trends, though non-significant, that suggesting that age of acquisition in either language might slightly increase the SCR. At the individual level, we found that the highest single SCR was 0.398 microsiemens in Hindi personal, and the lowest was English personal, 0.019. So we can see that while Hindi stimuli and personal context tend to elicit stronger responses on average, there was some individual level variability. But overall, yeah. Some did show a stronger reactivity to English stimuli or professional context. So from this study, we can sort of conclude that emotional reactivity in bilinguals is not only language dependent, but also dependent on how, like the context of the context in which it appears. So yeah, in the end, I would just like to conclude that, so when it comes to bilingual emotional language processing, context is also an important thing that comes up other than just language. Yeah. Thank you.
Thank you, Gori. We can now have some questions. Yeah.
Hello, Gori. Great talk. I just had few questions. I may have missed it. So, in your blog, in your design, is it possible that the words can be from any of the one of the situations, or are they grouped by blocks or something like that?
No, it was randomized.
It was random. Yeah. The second thing is, did you get these words rated for arousal and valence before you started?
No, not before I started, but in the end, as part of the questionnaire that we asked our participants to fill, we did get some feedback about the stimuli.
Because some words, if you in English and their Hindi counterpart, right? Even though they may mean the same thing, they may not carry the same importance, right? So maybe that is why, or the way they are pronounced and something like that could be seen in a GSR difference. So how do you account for that?
Yeah, so since our study was modeled after Harris's experiment, even in that experiment, what they did was that obviously we can't, like when it comes to two different languages, the meaning changes drastically. So even if it's like the exact translation, it won't, like the meaning will, is susceptible to like change. So there's not much that can be done to that, about that. But yeah, since we just try to make sure that, like we had the, like the counterparts, like the proper counterparts for the stimuli.
Yeah, so maybe set of criteria you could, so to avoid the as a confound, right? And last, sorry, were they having equal proficiency across Hindi and English, or is there a demarked proficiency for Hindi than English?
No, so the proficiency was self-rated, so we can't really have like an...
Yeah, maybe you could look at it in both order and see if you are still finding these effects.
Yeah.
Thank you.
Hi. So in one of the slides, you mentioned that the stimuli or the cues were limited to 5 words.
Yes.
So do you think that, can you give like for examples of what were the cues given to the people who are subjects? And if it was more than 5 words, like the context or the stimuli which was in the scope of this study, then there would be like deeper difference in the emotional processing in both L1 and L2 if we give a detailed or like elaborative context or stimuli to participants.
So the reason why we capped it to 5 words was because of the time limitations. Because like as we can see, Yeah, so since we had to like follow the temporal dynamic of the SCR as well, and we wanted to make sure that they are able to read and like imagine the sentences within the time. So we made sure that was the purpose basically. So if we had like if we showed them longer sentences, it would be harder for them to process those sentences within the given time. You don't have like a lot of time to sort of we have to make sure that the peak is captured and then the like the peak returns to the baseline and stuff like that, so that just to maintain the dynamics of the SCR.
Okay. Hi Gori, nice presentation. Just a couple of things. First is that it seems that the context only affects expression in the second language, not so much in the first language. Is that correct?
Yes, sir.
All right. And the other thing is about proficiency. So you have used LeapQ, which is self-rated measure. Did you consider using some of the objective measures like Lextail or dominance for that matter? Because probably language dominance given your task would be a better approximation of proficiency. So did you think about that?
Yeah, we actually wanted to do like a CERF test for proficiency or something like that, but yeah, because of time constraints.
All right.
But it can.
All right, thank you. Let us thank the speaker once again.
Thank you so much.
All right, thank you. Okay, so the third talk is an online talk. Yeah. And so this talk is by Madavi Manti from Kriya, and the title of the talk is Body Parts and Early Learned Verbs, Lexico-Semantic Networks in the Telugu and Hindi Contexts. Hi, Professor Maganti, are you there? Can you hear us?
Yeah, I'm here.
Yeah, so we are ready for your talk. Can you please yeah, share your screen?
Yes, yeah. So we have, okay. Can you see my screen?
Not yet.
Now?
Yes, yes. So you'll have 15 minutes. We can, you can please begin your talk.
Yeah, thank you so much. Thank you. I'll get started, okay? Yes.
Yes.
Yeah. Good afternoon, everyone. And at the onset, I would like to thank the ACCS organizing team for all the hard work, as well because this opportunity to present our work online in spite of all of these disruptions. Today, I'm presenting this collaborative work with two of my collaborators, Joshita Moin and Nitya Sethuraman from Grand Valley State University and University of Michigan, Dearborn. This work is titled Body Parts at Early Learned Lexico-Semantic Networks in Telugu and Hindi Contexts. So let's get started. Our framing question essentially concerns the role of body parts in verb semantics. By one position, body parts are an important scaffold in the early stages of development. Alternatively, the body and its parts are vital and pervasive for learning and organizing our conceptual and semantic knowledge across development. Let us see how our data provides evidence for either of these questions. But Traditional views of verb semantics have always focused on the conceptual features like path and manner, as you can see in this example, climb, which is most about path, but not about the hands grasping the drawers. So there's been very less attention paid to the body parts. But we cannot deny the ubiquity of body parts because body parts help humans interact with their environment. And it is through the body parts we take in the information. Body parts are also used in abstract and symbolic systems across cultures. We also use body parts for counting, for dance forms and sign languages. Above all, the first words across cultures relate to everyday activities with body parts. So we also have very good evidence from the neuroimaging studies where body parts are well represented in many brain regions, because there's quite some evidence from the adult neuroimaging studies, as you can see from this slide. But many of these studies come from Indo-European languages, and there have been very few studies with children. So body parts have been linked to the processing of concrete verbs mostly in adults, but very few in children. As you can see that we do have some data of body part norms with adults in different languages. And you can see the systemicity and coherence in associations between a small set of body parts and early learned verbs, including the abstract verbs. But there is no published work examining children and no systematic studies in India as such. So these results strongly suggest that body parts are important for verbs, but how do these findings fit with general theories of conceptual and semantic knowledge? Given that we have debates on conceptual knowledge and semantic organization in terms of the amodal perspective, which holds that language is a system of arbitrary symbols, where the mind and language is separate from the sensory-motor system. But alternatively, the embodied perspective is all about the views cognitive and sensory-motor system as interconnected throughout the lifespan. So it's important to acknowledge much of this, that's both of these different positions. So while the debate still continues, multidimensionality is another position that's making strides. Multidimensionality is a position where semantic knowledge is organized along the different dimensions, which include the sensory motor, emotional, interoceptive, and social experiences. But it is also something activated by the task as well as the context. So in this talk, we will discuss the data that provides evidence for the dynamic systems, and we will try to understand how the dynamic systems with task perception and individual differences, as well as multidimensionality. We use a semantic decision task to examine and how children connect verbs to body parts. We use judgments because in our previous pilots, we moved from associations because when children were asked to associate verbs, they provided us with more of the object responses. So judgments helped us to give us more holistic responses for the data. So these are the current questions that we have. We address these 4 different questions that are listed over here. In view of the time, I'm going to look into these questions with the data in the results section. So we compare Telugu and Hindi-speaking children's judgments. Coming to the method, we had 100 early learned verbs from the transcripts of mother-child interactions that we had, and these 100 verbs had both concrete as well as abstract verbs that were there. Coming to Hindi, we had about 84 verbs that were taken from the Hindi Communicative Development Inventory, which also had both concrete and the abstract verbs. 41 of these verbs were translational equivalents that were common between the Telugu and Hindi language. Coming to the participants, we had 57 children from Telugu, and we collected data with 2 geographical locations for Hindi at Asavarpur, which is a village in Haryana, and Hindi Pune data, which is an urban location in Maharashtra. And the children basically were from kindergarten and first grade. and they were basically from the Telugu and Hindi medium schools respectively. Coming to the prompt that we used for each of this, these children were individually tested by the native speaking researchers in each of these languages. Since most of you cannot understand Telugu, print out in English. So for the prompt that we used, what body parts do you use when you actually run or clap? And for the Hindi, we used आप किस शरीर के अंग के साथ खींचते हो. But for the Pune, we wanted to really see with the difference in the prompt or the task instructions could result in different kind of responses. So we used अगर आपको भागना है तो आप कैसे भागोगे. So these task instructions were slightly different. With respect to the procedure, there was a warm-up with each of the children. Then we also provided training verbs to help children understand this task. After the semantic elicitation task, we coded for the first body response. Children to respond to the local language, but we do see that some of the children did respond in English, especially in Telugu language, but with Hindi, Savarpur and Hindi, it was slightly lower. And we also coded this data for linguistic fragments to look into multidimensionality, also the gestures that were produced for each of these children, basically. So let's see how these results are looking like in our data, basically, in terms of how we understand. So in terms of the first question that we wanted to address were how systematic and coherent are Telugu and Hindi speaking children in their judgments. Across all of the 100 verbs and the 84 verbs with the two data sets in Hindi, we see that the responses were largely about the surface body parts, except for the mind, heart and brain. But with and also the top 5 body parts accounted for almost about 90% of the judgments across both Hindi and the Telugu data. But it was slightly different with the Pune data, where it was slightly lower, probably due to the task instruction. Even after we had excluded 15 children who provided with pure body parts, the percentage of judgments also was slightly lower than the other two data sets that we can see. So we can see some much of the systemicity and coherence from this data. Second, coming to how do body parts organize the semantic field of early learned verbs into semantic clusters, we used a correspondence analysis, which was a dimension reduction technique suitable for categorical data. And with the data which was all about verb and body part vectors, we do see that the variance accounted for by the first 4 dimensions in each of the language was somewhat similar, looking into how the data was almost systematic in each of these languages. So let's see how this data looks with each of the languages when it comes to the semantic fields. When we look into the Telugu data, you see that we have almost about 6 clusters that we can see. And these clustering of verbs and body parts, you can see that the ear verb basically is clustered towards the lower left quadrant, and the eye verbs and the leg verbs are clustered largely towards the most of the bottom right quadrant that you can see. And these are the verbs like go, walk, climb, fall, get down. And also that a third cluster is very much apparent in the top right quadrant that you can see over here, which is the hand verbs such as measure, cook, give, throw. And the fourth cluster of mouth verbs that are mostly linked to the mind and heart and the brain. And also that these verbs are also linked to the eye in some regions. So you can see that the hand and leg verbs there are also both hand and leg verbs over here, which is changing from verticality, that is from drive, take along, to horizontally in terms of lie down, go to sleep, and tilt. So let's look how this data is basically looking for the Asabarpo data that you can see. So with reference to that, you can see that these, again, we see the 6 main clusters over here with the Asabarpo data as well. But here you remember how the eye was clustered with leg in Telugu. But here if you see, this eye is clustered with mouth, mind and brain and heart here. Tongue is very clearly connected, there is a continuity between tongue. The tongue is clearly important and it continues with the mouth verb. Then you see that there is a third cluster of mouth verbs related across in the top upper left quadrants, where you can see these words like laugh, like bolna, batana, combined with the mind and heart over here, and along with there are hand verbs such as love, like, want, and which are also related to the I verbs that you can see. So you can see that these verbs like search, show, find, study are more hand and I verbs, and make, excuse, permit, and wish and want are verbs that are more with mouth, brain, and mind. Sleep and cry are about eye, mind, and mouth. So you can see a greater continuity between clusters over here in this data. But when we come to the Pune data, we also see a greater continuity between clusters. But here, instead of 6 clusters, we see only 5 main clusters, because the hand and leg verbs are on the same quadrant in the same dimensions that we can see. But you can see that here the distribution of is over 2 regions, especially tongue, lip, throat or mouth, and the neck, shoulder, waist and knee are with the limbs that you can see. Think is the verb that is mostly related to the mind and brain, and make, excuse, which is bahane banana, is with mouth. Search and find are basically dhundna and khojna are largely with the hand and eye. So you can see that in terms of looking into our third question of how robust is this structure developmentally, we see that across the grade level there was not it was not significant across all the 3 data sets that we see. But we also wanted to examine and look into this development cross-linguistically. So we used a simulation, which is a proxy for development, where we looked into how children who provided us with fewer body parts versus children who provided with more body parts compare across these clusters. And we also did another analysis of comparing the 41 translational equivalents across languages.
Sorry, we have just 2 minutes.
In terms of the overall structure of how the comparison is between the simulated developmental networks, I mean, this is an overview, but I'm going to show you separately each of these languages later. We do observe 2 dynamics that we can see.
Dr.
One
Madhavi
is
Lata,
the
sorry to interrupt.
segmentation that happens earlier with the less advanced children, and then we see a lot of recombination and integration happening in the later with the more advanced
can you hear
children
me?
over here. So you can see Telugu looks more integrated, and also the Pune data also looks more adult-like over here. So I'll explain each of this in the next slides more. But when we come to the 41 overlapping words that we can see.
Sorry, we just have 1 more minute left. Just wanted to 1 more minute, please.
Yes. We see a fundamental semantic field organization is preserved in each of this. We do see a lot of integration and recombination where I is combines with the ear and mind, with the mind and brain later in Telugu. But in Hindi, we also see that there are fewer body parts, how stomach and the hand verbs get clustered over here with I, and with the Pune data also you can see that there is an integration with a kind of integration and which looks more adult-like over here. So we do see a robustness of fundamental network where you can see that the semantic field organization is preserved, providing us evidence that verb body parts play an integral role across development. With the abstract verbs also we found 50% agreement, and cognitive verbs also were very opportunistic, like want, know, and providing more, where these verbs were mostly had lot of fragments. Children provided us with fragments and gesture, and we also had more of no responses, especially from the Pune data with respect to the cognitive and emotional verbs. How do body parts connect to the abstract verbs? These are some of the examples where the mouth played an outsized role in Telugu, but whereas in Hindi it was most about mind and brain in combination with hand that you can see. So what evidence does our data provide for multidimensionality? You can see that children also provided us with lot of data related with responses like gestures and fragments. The gesture and fragments across Telugu and Hindi data is somewhat at 17 and 16%. The proportion of the gesture data that was provided with Pune was substantially much more than two data sets, maybe because of the task instruction and influence of task instruction. We are yet to analyze this data. This is somewhat of the verbs of feeling and verbs of cognition, which may recruit pragmatic meaning and or linguistic context together with source or instrument, but overall concrete verbs do recruit most of these combinations. So we do see that our results provide evidence for multidimensionality and multimodal heterogeneity accounts for learning different kinds of verbs, including the abstract verbs and all of this. So our findings also connect to prior work on verb learning, processing, and teaching. We're also collecting data with Tamil, and let us know if you have any of them. So would like to thank all the children and teachers, and also special thanks to Nitya and Joshita, my colleagues who have shaped and contributed to the study significantly. Thank you one and all for giving us this opportunity. Look forward for the questions. Thank you so much.
All right, thank you so much. We have time for maybe one quick question. Questions? Comments?
Yeah, one question, Madhavi, about the consensus that you see among the responses. Can you comment a little bit on, you said that in abstract verbs, you did not see a 50% threshold on the consensus, but what about in the concrete verbs? Is there a body part specific consensus differences in terms of, I think?
Yes. Yes, sir. Actually, because I was going fast, probably didn't see that. But with respect to the concrete verbs, we see that these were largely light verbs, and these were verbs that had 50% association, largely with hand and leg verbs. It was the concrete verbs that we can see. But with the abstract verbs, we did not see the 50% associations, because these were the verbs that were also largely with linguistic fragments along with gestures and also no responses. So by we could see that fewer than 50% associations were there, and there was a difference between the abstract and the concrete verbs in some sense.
Last chance. All right. Case, thank you so much for your. Please let me.
Thank you so much.
Thank you.
Thanks.
Thank you.
And please let us know if there are any questions, because my student Sanya is over there, and she can address if there are any questions.
Sure, absolutely.
Thank you so much.
All right. Thank you. Okay, so we move to the last presentation for this session. This is, the title of the talk is Processing of Gender Neutral Four Names in Punjabi, and the speaker is Prof.
साउंड चला दे दीदी.
Good afternoon, everyone. I'm Prajapin Kore, and today I'll be presenting on processing of gender-neutral names in Punjabi. Following Adraja, I'll talk about my test case Punjabi, the method and design of the experiment, then I'll talk about the results, and I'll come to the discussion of the results, followed by conclusion and references. Gender, as we understand, is quite a complex feature of language, as it can be understood and viewed from various perspectives. Linguistically, if we see, it's a classification of nouns which can be based on certain properties. These properties can be semantic in nature, such as biological gender or animacy. It can be formal as well, such as morphological suffixes. Sometimes it's quite arbitrary in nature. And this gender classification gets reflected in other elements of the sentence that are associated with that noun. Cognitively, if we see, researchers have found that it's a fundamental lens through which people tend to see the world around them. And therefore, it's a sort of process of gendering by which we tend to ascribe masculinity or femininity to different entities that we come across. So, in general, there has been this thought that language, to some extent at least, shapes and influences the way we think and conceptualize. And researchers have found that this is true for gender as well. And therefore, there have been various attempts in recent times to make language representative for different genders. And in this line, there has been rise in many gender-neutral language forms, including pronouns, common nouns. These are some of the examples from different languages. Spanish, which has seen newly introduced gender-neutral morphemes, and then gender-neutral pronouns in English and Swedish as well. Not only pronouns and common names, even gender-neutral names are on the rise. And names, as we know that they are integral to our day-to-day language, and they are linguistically relevant as they encode important probabilistic knowledge. And gender is one such feature that names are known to encode. Coming to the processing of these nouns, we see that, first of all, since these are neutral, for the parser, they can be ambiguous as to whether the noun is a male or female. And literature so far has indicated that lexical semantic ambiguities are costly to process compared to is still a male preference even when the linguistic is neutral. For example, this study by Lasonde et al. 2013, they tested that how male and female pronouns, continuations referring to the antecedents in a masculine form and a neutral form, how these were processed, and saw that regardless whether the antecedent noun was neutral or masculine, he continuations were processed more easily. They took lesser reading times compared to the she continuations. However, there are few studies in which people have seen that the neutral conditions were processed similarly to the gender matching conditions as well. So there is no as such consensus how neutral nouns are processed, also because in all these studies the main focus was the stereotype mismatch, and the neutral conditions were more or less taken as control conditions. So therefore, we see that in general there is a lack on studies lack on how gender neutral nouns that do not provide any sort of cue to gender, these are processed. Moreover, we see that in Indo-Aryan languages, this is even scarce. And for proper names, we see that there are very little research. This is also because that in most other languages, names are gender specific. But we think that, okay, so in most of the previous studies, people have focused on common nouns, but proper names are referentially and semantically different from common nouns, so they might show some processing differences. And we chose test case as Punjabi language to investigate the processing of gender-neutral names, because unlike other languages, in Punjabi, gender-neutral names are much more prevalent compared to gender-specific names. So this is my test case. Punjabi is a SOV, it's a in modern Indo-Aryan language with SOV word order. So it's a grammatically gendered language with two genders, masculine and feminine, and it marks gender on verbs, some of the post positions, some adjectives as well. Most of the nouns are given, are ascribed a specific gender like masculine or feminine, but there is a subset of nouns which can refer to both males as well as females. So as I said, most of the names in Punjabi are gender neutral, and they are more or less combination of two words, and these names usually refer to abstract qualities. My specific research questions for the experiment was that how male, female, and specifically neutral names are processed for gender, and whether there is any specific gendered inference from neutral nouns. This is the method that I employed. So to select my stimuli, I did some pre-tests to choose the male, female, and neutral names. And my experiment was a non-cumulative self-paced reading experiment. I had 58 participants who were right-handed and who were native speakers of Punjabi, who had learned the language before the age of 5. These are my materials. So I divided my material into 6 counterbalance lists. Each list had 48 critical items and 96 fillers. Fillers remained same across all conditions. These are some of the snippets from my pre-test. I tested 150 names, and the participants had to rate the perceived gender of the name on a scale of 1 to 7, where 1 indicated definitely feminine and 7 indicated definitely masculine. This was the procedure that I used. The participants had to read the sentences at their own pace, and they pressed spacebar to move on the next word. Each sentence was followed by 2 tasks. The first one was acceptability judgment task, and the second one was probe task for attention check. This was my design. It is a 3x2 factorial design wherein I manipulated 2 factors. My first factor was gender of the name, which had 3 levels, neutral, male, and female. And the second factor was gender agreement at the verb, which had 2 levels, masculine and feminine. This resulted in 6 conditions. So as you see, in the first 2 conditions, it's a neutral name followed by masculine verb or a feminine verb. In the third and fourth condition, it's a male name followed by masculine verb and then a feminine verb. And the last 2 conditions, it's a female name followed by a masculine verb and a feminine verb. So NM, NF, MM, MF, FM, and FF, these were my conditions. And the hypothesis was that at the position of the noun, that is the name, I expected to see longer reading times in the first 2 conditions, that is the neutral conditions, compared to 3rd, 4th, 5th, and 6th. This is because as the previous literature suggests, these would be ambiguous for the parser. At the position of the noun, verb, my prediction is that for the last 4 conditions, that is MM, MF, FM, and FF. So these are gender-specific names. So in the incongruent conditions, that is MF and FM, I expect to see longer reading times compared to their control paths. For the neutral conditions, I have 2 hypotheses. My null hypothesis is that I do not expect to see any difference between the neutral conditions and the control congruent conditions. And my alternative hypothesis is that I do expect to see a gendered inference from the neutral names, such that in the first condition, if it is inferred as male, then I should expect to see longer reading time at the position of the verb in NF condition. these are my behavioral results, wherein I saw that for all the conditions, overall acceptability was high, as you can see in the table. And for the neutral condition specifically, although neutral masculine condition was rated a bit higher in acceptability, however, it's not a really significant difference. The probe accuracy was also high across all the conditions, meaning that the participants were attentive. This is the plot for mean reading times. Here we can see the pink line and the blue line for neutral names. They are taking lesser reading times at all three positions, noun, verb, and auxiliary, compared to the gender-specific conditions. So this was really surprising result, as instead of their ambiguous nature leading to longer reading times, we saw that neutral names are in fact being processed much more easily. They are taking lesser reading times. So as I just said, we saw only the main effect of name type on all the relevant three positions, and it was highly significant. We did not see any effect of agreement type or any interaction. And a post hoc comparison compared that it was in fact the neutral names that are taking the lesser reading time. These are the plots of post hoc comparison. So here at the noun position, we see the neutral nouns are taking lesser time compared to the male and female both. And at the verb position too, we see that it's the neutral conditions that are taking lesser reading time. So what we see in our result, we speculatively interpret that maybe since in the language the neutral names are so frequent, so their high prevalence in the language is making people easy to process these names compared to the gender-specific ones. And further, since we did not find any effect of agreement or interaction, that means there was no gender-specific inference from the neutral names as well. And what is even more surprising than that, even in incongruent conditions wherein there was a specific gender name but followed by a mismatching verb, there also we did not see that they were different from the congruent conditions. This we interpret that this long time exposure of the names in the language and the usage of these names along with frequency is making people, is influencing the processing of gender-specific names as well, such that people are not readily committing to any gender-specific inference at the noun itself. However, we do see a general preference for neutral names reflected in the shorter reading times. However, this speculation needs to be tested, how frequency is interacting with ambiguity in further experiments. But however, this was an attempt to understand how gender-neutral names are being processed in Punjabi. These are my references. Thank you. I'd be happy to take questions.
All right, thank you. Questions? Yes, we have a question.
Hello. So in one of the slides when you were telling about the participants, it was mentioned there were 58 participants which were right-handed. So right-handed, left-handed, does it play any role in this scheme of work? And second, there are 4 names which can be gender neutral across one dialect, and it can be not neutral across the dialect of the same language. So was these taken into consideration, like it can be gender neutral for one dialect of the same language and not for another, because yeah.
Okay, thank you for your question. Regarding your first question, my reason for taking right-handed participants, although because people have seen in language processing studies that left-handed and right-handed people might differ in the way they process language because of the lateralization. And regarding your second question, if I am interpreting it correctly, you are saying that in the same language, in one dialect, the name may be neutral and in another, it might not be neutral. But the names that we did, we test to choose our names. So, and these participants were not just specific to one dialect. It was a very representative sample in that sense. So, yes.
Can I ask a question? So, can you go to your stimuli, please?
Yes.
So, if
so design?
Yeah, design. Yeah. No, the stimuli.
Right.
Yeah. So, if I understand correctly, you're saying that if you have a stereotypical feminine name, and then you have a verb which does not agree, then you don't see an effect?
Yes.
That should be a red flag, no? Why should I not see an effect?
Exactly. That we expected, that when a name is being rated as masculine, but in the self-paced we did not see a mismatch effect. And this is like, we are still in the process of interpreting, but
Are you worried about this not appearing?
I wouldn't say worried, because in some sense it can be positive as well. One thing is that gender of names, even if they are rated as specific, it's still probabilistic in nature. It's world knowledge based gender. Yes. So if, yeah.
So I just wanted to flag two things. One is that when you measure the reading time at the verb, correct, your pre-critical region is distinct across the different conditions, you know. So you could potentially, and self-paced reading is known to have spillover effects. So that's one point. The other quick point that I had was regarding the neutral condition. So there you're seeing an advantage, right? And the advantage could potentially be explained by the fact that in the neutral condition, there's ambiguity, right? It could be either. And the parser basically chooses one, always has an interpretation, unlike the other conditions. So there's some kind of an ambiguity advantage that you're getting for neutral compared to others. And garden path literature, if you look at that literature, you will find that there is a attested ambiguity advantage effect that is seen, where ambiguous instances can actually be faster because there's always one interpretation available. So that's another way in which perhaps you could interpret the advantage of the neutral sort of conditions.
Yes, so I might be wrong, and this is actually like I mean, you're right in that sense that in garden path studies, people have seen that ambiguous sometimes are read faster. But isn't it the case when it's shallow processing and when deeper semantic process? I don't know what is really happening here. I'm still in the process of interpreting.
The thing is that in the neutral condition, you always have one interpretation potentially. A reader can either go for masculine or feminine and can go with it and interpret the sentence accordingly, is what I was trying to get at. But
right. So just I would like to know more. In that case, should we see if there is one inference, but if that doesn't matches at the position of the verb, we should see a longer reading time?
I suppose, yeah. I mean, the reading time at the verb should appear. Yes.
A related comment to Summer. What were the acceptability judgments for those where the noun and the verb were incongruent?
Yes, I'll go
I mean,
to
there
that.
should be a difference, right?
There
You would
is,
expect not
yes,
just in reading time, but also in the acceptance.
Yes, I would like to talk about it. So if you see that in the incongruent condition MF, although comparatively all our acceptability ratings are high, but you see a drop in the MF condition. That is a male name followed by feminine verb compared to the MM, 85 and 74. And this is also statistically significant. But if you see this plot, this is the EM means plot of acceptability ratings. So we see that these two conditions are different. However, they are not really different in a meaningful sense because the acceptability drop is not something like 25 or 20. That is the usual pattern in the incongruent conditions. So yes, we do see a difference, but it's and also only in the male condition. So in the female names condition, you see that there is no difference, even in the acceptability. So yes, there is a difference, but it's not really a meaningful one.
Hello. Sorry. So can I ask a question now?
Yes.
Very briefly. So thank you for the novelty of the work. My question was regarding the implications of the current work in a theoretical perspective. What are your thoughts on this one?
Right. It's like currently people are investigating, since most of the languages are seeing intersection in neutral forms. But in other languages, these are recent inventions. So people have seen higher processing costs for such names, nouns, or pronouns compared to the traditional ones. But we wanted to test if with more exposure, like some people have suggested that gender neutralization as a strategy for making language gender fair, that can be effective if there is more frequent use and exposure, just to test that if that works. But it seems to be working well in this case. So experimentally, yes.
No, most of the results which are obtained here can be driven from the familiarity as a factor. Very simple factor, right?
Yes.
But what is that which makes this work so interesting from a theoretical point of view, I'm asking. But it's fine, I get your idea also.
Okay, I think we will have to end. Thank you. I'm sorry to sort of, we can take this, after in the break, so please. So let us thank the speaker. Thank you so much. And this ends the session. This brings us to the end of this session, and so we'll have the keynote now.
There's a small change in the schedule. We have the keynote right now, followed by the tea break. So it's my great pleasure to invite Professor Bharat Biswal as our next keynote speaker. He is professor at the Department of Biomedical Engineering, New Jersey Institute of Technology, and a pioneer in the field of neuroimaging, specializing in biophysics, medical imaging, and cognitive neuroscience. And he's credited with developing the technique of resting state brain connectivity. Sir, we all look forward to your talk. I invite Dr. Bapi Raju, IIIT Hyderabad, to chair this session. Thank you.
Standing. So I think, thank you for the introduction. So I think most of you would have heard rest, brain never rests. So the credit goes to the pioneering work from his group, that we know so much about resting state, and the fact that you would always think this is a baseline, you would factor it away, and I'm into the task. All that paradigm has changed, and a lot of work on how to use this meaningfully to characterize a variety of neurological disorders, psychiatric disorders, developmental disorders, lot of things that have happened using this as a signature. So I have the pleasure and honor of chairing this session. Invite you to give you a keynote talk. Thank you.
Thank you, Dr. Raju, for the nice introduction. I would like to thank, can you hear? Okay. I would like to thank Dr. Bhumika for inviting me back again, and the organizing committee for letting me come back. Also good to see so many old friends. So I'll be talking about will it be better? I think it's okay. It's okay. So I'll be talking about maybe a little bit of rest, but more about movie watching today. So for those of you who are new to fMRI, so with MRI we can get high resolution images with maybe 1 cubic millimeter spatial resolution. With fMRI, you can collect a spatial resolution about 3 millimeters. But in this case, what you can do is you can collect images at a much faster rate, right? So you collect images every 2 seconds, so you can make a movie instead of a digital picture. And the basic idea for fMRI is based on bold signal contrast, which suggests that when there is when you perform a task, there is increased neuronal activity, which leads to vasodilation, and because there is more oxygenated red blood cells compared to deoxygenated red blood cells. So therefore, there is an increase in the bold signal. Right. So this is one of the first studies of fMRI, where using a flashing checkerboard, you have baseline, then you show this, this is blank screen, then you show this flashing checkerboard, and then again there is offline, right, dark screen. So when you show the flashing checkerboard, in the visual cortex, the signal goes up. When you show like a blank screen, the signal comes down, and then again when you show the flashing checkerboard, the signal goes up. And this is just representing the same thing in a paradigm form. Like you have blank screen, intact object, blank screen, scrambled object. And then you can see when, well, during blank screen there is less signal. During intact object there is, there is an increase in signal activity. During blank screen, again the signal comes down. With scrambled object, not so much, but still some activity, and so on. And there are many, many different, I should say. So if we want to find differences between two conditions, we can use some standard statistics to show differences between two conditions. And then the voxels that survive, the threshold, you say, okay, when I do finger tapping, or when I see a visual stimulus, the visual cortex lights up, or specific brain regions lights up. And by doing more complex tasks that was described earlier today, you can find very specific brain regions like object, faces, and places, and then you use the same kind of design to analyze the data and identify those brain regions. So the idea with fMRI task activation is you present a block design, and then you have generally a control condition, and then you use so this is the block design, this is your actual response. There are many different ways in which you can analyze the data. It's like t-test or correlation, like GLM is the most common one used. And then you get statistics on a voxel-wise basis or a region-wise basis. And then what you say is that, okay, voxels that pass a specific threshold that you set, those are activated voxels. You say that, okay, when I do this task, this region is activated. So the other way to do brain imaging has been to look at the baseline condition, and that is the condition during rest. So this is looking at the frequency spectrum. So this is from the gray matter. This is the heart rate, harmonics of the heart rate, respiration, and then this is the low frequency signal, and that there is a difference between gray matter, white matter, and that the noise characteristics outside the brain is much less, right? Like this one. And so what I had done for my PhD was analyze the resting state data, very similar to the way we do the task activation data. So this is voxel from the sensory motor cortex during rest. And so we took that time series and used like a GLM approach, right? And then while this is from finger tapping experiment, this is just during rest, right? Since there are many students here, my paper got rejected from 4 journals. It took me 2 years to publish. So, if your paper gets rejected once or twice, it's nothing. So this is like basically resting state connectivity. And because Carl Friston had defined functional connectivity as correlation between physically distant region, and that we were observing this during rest, we defined it as resting state functional connectivity. Again, task activation has many, many advantages, but we have to admit there are many things task activation cannot do. And one is that in resting state, from one single resting state scan, you can identify many different resting state or many different functional network. You can identify the sensory motor network, you can identify the visual, the language, executive, and so on, just from one resting state scan, right? And again, this is a study using more than 1,000 subjects. And then in here, what we show is that from 1,000 subjects, where the data was collected using different scanners like GE, Siemens, Philips, 1.5 Tesla, 3 Tesla, 4 Tesla, and 7 Tesla, different pulse sequences, different countries, we could identify like distinct networks, like distinct resting state networks. And again, because we had so many data set, we could look at, for example, gender differences or age-related differences. So another maybe limitation of task activation is that for many kind of population, you simply cannot perform a task, right? So this is a study in infants where just from the resting state scan, we can identify the sensory motor cortex, visual cortex, the default mode using seed-based analysis where you take a time series from a specific brain region and correlate with rest of the brain, or using another method called ICA analysis, right? Independent component analysis, you can identify them. Further, you could also look at differences between children and adults, and then show differences between the two groups. And this is work by Damien Fair, where they are showing differences in children from the network level versus adults. Like in adults, many of these networks have been formed, which is still developing in children. One can also, as Dr. Bapu mentioned, look at different diseases, like, and this is depression, and looking at depression versus healthy control. And here what is shown is that there are regions where there is hyperconnectivity in depression compared to healthy control, as well as regions which is hypoconnected compared to healthy control. So some regions are more active and some regions are less active, right? And again, I would argue that to do such a kind of task is not very simple, not that easy in task activation. This is like one of my favorite study where Helen Zuo and colleagues decided to look at not versus healthy control, but different types of neurodegenerative disorders, Alzheimer's, frontotemporal dementia, ST, and two other disorders. And then both sensitivity as well as specificity, right? So we have features which are unique to Alzheimer's, and that we can use this feature not only to differentiate between Alzheimer's and healthy control, but also to differentiate between Alzheimer's and frontotemporal dementia and other kinds of disorders. And as I've said, with resting state, there has been a lot of criticism by people doing task activation. And this is the editor-in-chief of Neuroimage, who wrote this paper, Does the Brain Have a Baseline and Why We Should Be Resisting Rest? And their main criticism was, I would really suggest for students to read this paper, because it tells you what they really think, right? Like, it's good to have arguments sometime with friends, because then they tell you exactly what they think about you. So in this case, the point was that why should we bother doing resting state? Because most of what we know about neuroscience has been obtained from task activation, so why bother, right? The other criticism has been that how can we correct for resting state when we don't know what the ground truth is? You can always try to filter out or remove one kind of artifact, but since you don't really know what the signal is, you can only try to minimize the artifact, but never correct. And then the third one was like, they would believe, okay, this paper was published like in 2007. At that time, most of the fMRI study was done using 20 to 30 subjects. So they said that, they would only take resting state seriously if a paper was written using 500 to 1,000 data set. That's how I got the idea to do the 1,000 connectome, right? But recently there's been other people who have criticized. Again, like the thing is, some of them write so well. I wish I could write like that. And again, here they criticize that you should not do. Now they're saying that, oh, it's a good method, but it's past its prime, so we should, move on from resting state. So what I would like to say is that maybe now there are three kinds of paradigms. One, of course, resting state is my favorite, but the task activation is another one which is very widely used. And there is a third one which has emerged called naturalistic paradigm. And the naturalistic paradigm has been particularly of interest to me because people who have used naturalistic paradigms are using the analysis methods developed for resting state, but sometimes they're using these methods, they're using naturalistic paradigm then to criticize resting state, right? So I thought, okay, I should investigate this naturalistic paradigm. And so I should have said, so the naturalistic paradigm actually is the first paper that was published by Yuri Hassan in 2004, which was published in Science. And what they did was very clever. They made a bunch of subjects watch the same movie. It's a famous movie called The Good, the Bad, and the Ugly by Clint Eastwood. And this is like looking at specific brain regions and across 5 subjects. And then they showed that the temporal pattern across different subjects was the same. And what they showed was that, like, this is the fusiform gyrus, right? So every time, like Clint Eastwood or the actress face appeared, there would be like an event, right? There would be the signal that they saw. Similarly, for other scenes, they could make this action, right, corresponding to different tasks, right? And they showed that this was highly predictable across subjects, right? And so what naturalistic paradigms people have been saying is that first, that, this is a better stimuli compared to like task activation, and it simply involves watching videos or movie or listening to music, and it represents our more day-to-day activity than performing like a flanker task or tapping your fingers. And that for some complex, like for block design, there can be habituation and the other effect, but because you are watching a series of events, for some complex function, it may take time to build up, which cannot be done, or it cannot be easily done by a block design, and therefore naturalistic paradigm is a better way to look at the data. And so in this case, so we used a data set which had resting state, then it also had Inscape. Inscape is like a three-dimensional feature with simple music, and it doesn't require a lot of attention, and you just have to see. And then they also showed several different kinds of movies. And so the paradigm, so the other advantage of this movie watching or naturalistic stimulus is that there's a few papers now by Tamara van der Waal, where they show that, like, both for Inscape as well as movie, like, you can minimize head motion, especially in children, right? In children who tend to move a lot, if you show them this, like Inscape or like movie, they don't move their head so much. And also the amount of drowsiness reported is less, right? So this is what they reported, that head motion compliance, like movie watching had the least amount of head motion, Inscape had slightly more, and rest had the least, right? So, and they said was that movie watching, they could use 79% of the data, for Inscape 77, while for movie watching, they could only use 44% of the data. So one thought I had was that if movie watching is so good, should I just stop doing resting state and move to movie watching? Or could it be that movie watching is completely different? Because, like, I don't know about you, I have seen some video clips which have made me feel uneasy even an hour later, right? And so if, like, is movie watching a resting state condition or something else? So that was the primary objective, that is resting state that we get from scanning someone at rest, the connectivity pattern that we get, is it different than what we would get when someone is watching a movie? So we wanted to look at spatial patterns during resting state, Inscape, and movie watching, and we wanted to see the temporal consistency during Inscape and movie watching. Also, we wanted to verify, as I said, if functional connectivity of Inscape or movie watching is close to the resting state, because if it's close to the resting state, maybe, I should start doing something else. So we use this data. This is a publicly accessible data, which was done by Tamara van der Waal. And so what they did was they had 13 healthy subjects between 21 and 42 years of age, 7 female, 6 male, and so what was done was each scan was 10 minutes, and there was 4 tasks per condition, and each subject was scanned 12 sessions, right? So each subject was scanned on 12 different days, right? So there was a lot of movie data that was collected, a lot of Inscape data that was collected, and a lot of resting state data that was collected. And for this study, because I was just interested on movie watching and Inscape versus resting state, although they had a block design flanker task, we did not use it. So this is how a typical scanning session looked like. So this is the flanker task, resting state. There's a movie called Volley, then Inscape, then again resting state, Inscape, flanker task, A Few Good Men, then movie Matrix, flanker, Inscape, and so on, right? So this is for one session, and there were 12 sessions like this, right? So this is the pre-processing that was used, and the pre-processing that is used is almost identical to the way we analyze the resting state data, right? So standard realignment, co-registration, segmentation, motion correction, and regression. In here, though, we tried to do one additional step, and that was because everyone was watching the same movie or Inscape had the same sequence. What we did was, so let's say there was, again, this was done for both within subject as well as across subject. So let's say Inscape was, the data was collected on session 1, but there was Inscape that was collected across session 2, session 3 till session 12. So what we did was, on a voxel wise, we averaged this, right? So if you're averaging 11 sessions, so we should get something that represents only changes due to Inscape, right? Because the only thing that is common is the presentation from Inscape. Is that clear? Okay. So then what was done was we regressed that out, right? Because it's like, I don't want to know just what happens when you're watching a movie. That's very well known. But what I'm interested is, if I can extract the response from Inscape or the specific movies, then what is left is that resting state, because theoretically, that should be the resting state, right? And similarly, we did this for the different movies, right? So as I mentioned, first we used ICA for rest, Inscape, and movie. Then we used tensor ICA. Like tensor ICA is another form of ICA where the temporal components is the same across different subject or different sessions. So you can then specifically bring out those components, right? So like in ICA, for resting state, concatenation is used, while for task activation, because you have specific features, the tensor ICA is used. So we analyzed it using both ways. And then we also looked at group theoretical measures like correlation matrices for these data. So we wanted to see the differences between the different conditions, right? And these are the different networks. that has already been mentioned, the visual, dorsolateral attention, and so on. So this is from one, for all subject from session 1, right? So these are, again, most of the resting state is done on like 20 or 30 subject. This is only 12 subject. I haven't done any pre-process, like any post-processing, any clustering. So this is the raw data, right? So you can see the medial visual, the sensory motor, the dorsal attention, right? The 7 networks, the default mode, and so on. Right? And then this is the inscape that we get, right? And this is the different networks. So we see very similar networks as we see for resting state, but when we regress it, then these networks are not as reliable or not as strong as we would see in resting state. Similarly, we did for movie watching, and then we did the regression, and after regression, we can only find these 4 networks, right? So again, the point is, when you are watching a movie and then you're extracting the component corresponding to the movie, then the network that you see is not what you see in the resting state network. This is the correlation pattern across the resting state network, across the 12 subjects. Again, I haven't done any processing. It's just like, from the first session for the 12 subject, there is, some variability across the subjects, and that we can find out. And then when we do for Inscape, we see also a pattern which are similar, right? And then when we look at resting state versus Inscape, there are some similarity, but there are also differences that I'm going to cover later, right? So this is for movie after regression, and these ones are the different networks, right? So this is voxels from the sensory motor cortex, right? Like from the default mode network, the visual, memory, right? So these are the different brain networks. So everything is correlated, everything, and therefore the diagonal is correlating itself, and therefore is high, right? And when we look through these differences, we can see that many regions which are quite different compared to resting state. So this is resting state. Again, this is for movie watching versus resting state. And then this is again movie versus rest for the different network. And there are many regions which are significantly different, right? Again, these are differences between movies and rest. So basically, spatial ICA reveals more networks for Inscape and movie compared to rest. All 6 networks from rest with additional occipital and auditory network, right? So resting state had this additional network that Inscape and movie did not, right? So activation in more functional network during natural viewing. So while there is more activation while watching movies, but when you regress these out, the connectivity pattern that you get is very different from what you get during resting state. Right? So right? So again, higher correlation in visual attention, salience, and parietal network. So this is like movie versus escape and rest. Indicates demand in cognition, emotion, and attention, which is movie is greater than escape, then greater than rest. Again, this suggests that you can get some network structure, but these networks are not what you get during resting state. And so one has to be a little careful. Again, so this is just showing pair t-test for escape versus movie after like, that was 35 versus 61 with significant differences. Between escape versus movie after regression, it was 41 versus 73 ROIs. So they demonstrate less differences with resting for escape, while for movie watching, there is significantly differences. So then the other question we wanted to ask is that, you know, when you're watching a movie, there is a sequence of events that occur, right? And as was mentioned in the, I guess, one of the first talks today, that the brain connectivity is time varying and it's not stationary, right? So with like typically the way we had done connectivity analysis is we would take 2 time series, find their correlation and say, okay, this is the correlation between 2 time series. But a lot of work by Katie Chang and others have shown that these connectivity patterns vary as a function of time, and one can do like segmented, like you do correlation between 2 time series for a block, then go to like a next segment overlapping, or else you can do non-overlapping, and then you can find correlation coefficient changing as a function of time, right? So this is like, so you can do correlation using a time window across the entire time series and see how they change as a function of time to look at the temporal variance in this correlation structure. And this is showing correlation between different brain regions across different subjects. And this is the average response for window. So now what we can see is specific pattern between different brain regions while they are watching the same movie, right? And this can be used to look at the correlation structure or the time varying structure for 3 different movies. This is Wall-E, The Matrix, and A Few Good Men. And this is showing how is the structure different between Wall-E versus these 2 movies, The Matrix versus the other 2, and The Few Good Men versus the other 2. And this is showing their temporal structure, right? Like which regions, which independent component region are connected or change temporally while someone is watching a movie. And this is looking at these connectivity pattern across the different independent component for the 3 movies, and then looking at the differences between them, right? So then what we wanted to look was that what is the difference between the time varying correlation structure versus the stationary connectivity that is obtained, and is the time varying connectivity more sensitive to movie contents, and if Could it be that time-varying connectivity is more functionally relevant? Also, can these reflect some individual differences of behavioral phenotype, right? So this is using the Healthy Brain Network, which has like close to 300 subjects. And in this case, they would watch 2 movies, The Present and Despicable Me. And in addition to age and gender, we also had FSIQ and SRS scores. Right? So we use the standard analysis methods, and these are the 18 functionally relevant independent components. And because we wanted to model the age, so this is like a constant parameter. We wanted to regress the site effect, the gender effect, frame-wise displacement, the translation and the rotation part. And then we used, like, one model was to use age as a factor, then use quadratic age, like different age effect model we used, right? And so this is what we use. So for every subject, we have regional activity, we have stationary connectivity, then we have time-varying connectivity, and then we modeled this to the different factors, right? And then we had the observed, and using these measures, we also tried to predict, right? Right, so this is showing the independent component analysis, basically what fraction of the percent, how much variance could each component account for the 2 movies. And then this is The Present, this is Despicable Me, and then this is the time-varying component, right? And these are the components that we used, the 18 components that we used. And what we see is that by using a Chi-K weight, we could see how different components could account for age by using like different independent component could account for age. Like for this one, using the first principal component loading, you could fit the age and try to model with some prediction how good a model it would be, right? Then this is again using the time-varying connectivity, looking at like how good a fit this was. And so this was the Present movie, while this is the Despicable Me movie. And then this is using stationary connectivity, again for the different independent component pairs, because there are 18 components. So it's 18 times 17 divided by 2, the different components, and how the different weight contributes, right? So some contribute highly, right? Some not so much. This is using the dynamic connectivity pattern. And then this is again showing the regional effect, right? So this is stationary connectivity, stationary connectivity for the Present, and then principal loading between the ICA and the 7th ICA. And so basically by using this kind of analysis, what we can do is we can basically with some prediction tell from their fMRI response we can predict someone's age by using this kind of analysis. We can tell whether it's a male or a female. Then we wanted to see whether we could use this information to make some behavioral prediction. And this is where we, I thought like I would get better prediction with time varying connectivity, since I could get better prediction about age and gender using time varying connectivity. But that was not the case. It seems that the stationary connectivity had better prediction. In this case, it was 0.347, while with using time varying connectivity, it was 0.138. For Despicable Me also, it was about 0.348, and the time varying connectivity was minus 0.214. For the SRS, the results, there was not much difference, in a sense that this was 0.14, this was minus 0.15, so no real difference. And while this one was for Despicable Me was 0.3, while with time varying connectivity it was minus 0.2. So to summarize, so different age effects, there is different age effect between time varying and stationary connectivity, but especially for FSIQ and SRS, I don't know how to summarize, because it doesn't like stationary connectivity seems to be a little better, but for age prediction or gender prediction, time varying connectivity seems to be better. So maybe stationary connectivity had better prediction performance. Maybe stationary connectivity is more trait-like, while time varying connectivity is more state-like. That is my only interpretation. Maybe with movie watching, maybe it's good. Like if you can scan subject, like children, by watching them, making them watch a movie and they don't move, I think it is good. But I think we cannot simply say that it is resting state connectivity. It is maybe a different connectivity which is corresponding to the movie that they are watching. And perhaps that is all we can say. Again, there's a lot of work to be done. We are looking at whether we can come up with other models like hidden Markov model to better model this. Last but not the least, most of the work that I presented was done by Shin, Pratik, Sapna, and Sukesh. If you have any questions, I'll try to answer. Thank you.
Yeah. I have a question.
Hello, sir. Can you hear me? Great talk, sir. So, it's very interesting talk, Professor Biswal. So I am very curious about how did you select those three different types of movies? What was the rationale behind it? So for example, if you compare Matrix movie with Wall-E, I can see a lot of differences. Like for example, Matrix is very science fiction movie and it is very action movie, right? But not Wall-E, but I can see some similarity. Like here, both are exploring humanities. It was trapped by technology, robots, and so on. Right. So, I mean, I can see you also found some differences, across movies too. So what are those rationale, like choosing these three movies?
Right. So actually, the three movies that you mentioned, those were collected by Tamara Vanderwall, who was doing a pediatric study. So The other two movie that I mentioned, we collected those movies. But to answer your question, that is a very difficult question, right? What movies to watch to show the subject, and that almost depends on what is the question you're trying to ask, right? You can, I have thought of giving subjects maybe Charlie Chaplin movies rather than like Clint Eastwood movie, but that would be to perhaps a different kind of subject. For children, generally, people have used, for some reason, cartoons, but I guess like children somehow like cartoons more. So, so again, I wish I knew how to answer your question better, but yeah.
It generates different emotions.
Different
movies generate different emotions.
Right,
and it can affect the results in terms of brain connectivity and so on.
Right, and I think I have mentioned it, I have mentioned it that, you know, if I was to find differences in men versus women, right, I can think of several movies versus if you wanted to find differences between young and old, maybe, you know, I can think of another set of movies. And so I think that's, there's couple of studies that have been recently published, like trying to answer that question, like even it's so like dependent on the geography and culture that, you know, what movies to show who could be a whole study by itself. Yeah. So there's been some study where they're also having the subjects rate the movies as they're watching, so to get additional parameters to use it as a correlation.
Great talk, sir. So I had a question. This was coming from an aging perspective. So we usually take resting fMRI in terms of aging, right? And we try to look at scaffolding or compensation networks in that. Right. But now that after the talk, I was thinking when we are looking at from a movie perspective or from the in-space perspective that you are talking about, right. Do you think that this would be a better paradigm to use and look at compensation? I mean, I just wanted your views. And should I ask my other question as well?
Okay, so I do aging work using resting state. And I would say that you can, and I don't think there's many study. I think there's one or two study where they have looked at young versus old while they're watching movie, like the CAN study is there. So there are a few studies, and so you can do that, but I would say that also collect resting state, because with aging there is assumption that most people make, and that is that the underlying vasculature is the same between young and old. That is the assumption people make, which is not true, right? But if you're doing resting state, you can easily identify that, and then there are ways to calibrate the responses. So you can do movie watching, but also do resting state, and then calibrate your responses.
Hello. Hi, Bharat.
Hey.
Good to see you. So, just a Q. Oh. Okay, I go to see it. So, the part is good. No, please. After you.
Just Michael Jackson.
Can you hear me? Yes. So I think one take-home message from this your presentation is that the movie watching data is different from resting state data, and they have like different information and all this. But even if we consider resting state data, and who work with neuroimaging, we know it's very much dependent on the method that we use. So for example, if you used, I think, functional like connectivity, correlation, but if you use Granger causality or dynamic causal modeling or something else like mutual information or any other method, we'll probably get completely different networks. There's some connection. So my question is how you see this, like how they are related, even philosophically. Different networks that we get from different measures of functional and effective connectivity. Even for the dynamic, probably you use sliding window dynamic connectivity, but if you use, say, HMMR or something, in my experience, we'll get completely different types of pattern of like states and all this. So this is, and how they are connected, so like the different networks that we get from Granger and DCM and from the same data using different methods.
Right. Yeah, that's a very good but difficult question. So I would say that there is still similarity, right? If I'm using correlation analysis, my seed is in the sensory motor cortex, I will find the sensory motor network, which I will find using ICA, which is what I will find using PCA, right? Now, maybe I shouldn't talk about DCM with him here. So maybe for Granger causality or DCM, that I don't think the question that is asked is what is connected to what. I think with dynamic causal modeling, it's a different question that is asked, right? So I would say that with resting state, the results is fairly consistent that, the main finding that, the sensory motor cortex, even at rest, is connected, right? The visual cortex at rest is connected among themselves, and other networks also. So that is the main finding, and regardless of the methods that we use, we get similar results. Now, so that is what I would say. Was there a second question you asked? Okay, thank you.
Thank you. Hi, yeah. So, I mean, fantastic talk as always. So, just a comment, more of a comment. So, in our kind of, a lot of the analysis that we do with this dynamic functional connectivity kind of approaches, when we particularly look at it from the perspective of these time-by-time correlations, there we do see, very different kind of patterns. From rest to movie. So to cut the long story short, rest seems to be more where you are going through these repetitive states, right? So DMN and SMN, all that, versus the task where things become very low dimensional, which is why what I think Dipanjan was trying to refer. We use these unsupervised approaches versus the sliding window approaches.
Right.
And I think it may be reassuring more to you. So rest is very different from the movie. Right. At least that's what we find. Right. And I think those dynamic functional, which you showed. I mean, you showed the correlations in a time-dependent fashion. Those are, collapsed across the all time, right? So that's one disadvantage. Right. So if you kind of, unpack that in time, right. I think those differences get more drastic between the rest and the movie.
Right. Yeah,
that's it.
Yeah, thank you. Yes.
Thank you so much. This was really, really useful and interesting as well. So my question is, just so I understand, are you regressing in the movie watching, are you regressing the stimuli out? Are you regressing that out or not?
So I'm regressing the response due to the movie out.
Out, right?
Yeah.
Okay.
Yeah.
Okay. And so in these sort of head-to-head comparisons, right, with REST and that, do you find sort of qualitative differences in the context of, say, the strength of connectivity within network, right, or cross-network? Are you seeing things like that differ?
Right. So we do see a lot of differences. Maybe I should have shown. So what I was showing was that using resting state, let's say we got 7 networks. Again, with the small number of subject, just using one session, we got 7. But when we regress it out from Inkscape, we would network, which, but for the movie watching, we would get 4 networks. The other ones were much smaller, right? So why that is happening? Maybe the signal that we are regressing has those components, or the signal that is generated is somehow like a Steele effect. Those signal from those regions are the main contributors, and therefore we don't see any meaningful correlation after that.
Or perhaps the movie state, potentiates some networks, right? That are more relevant.
Right.
So from sort of these comparisons, do you have any recommendations for network specific? So let's say one is interested, because of your research question in the sensory network, say visual or whatever.
Right.
What state may be better? Like I would imagine that visual would be better, seen in the movie watching. But what about these? So say default mode, control networks, these higher level networks.
Right.
Do you have a sense like what would be better as a state to use, REST versus?
I don't really know. I would have.
I have two brief questions. A privilege of being the chair. I'm not familiar with this Inkscape movie.
Right.
Does it also have? Is it audio visual like movie?
So yeah. So.
And so that you see the occipital and auditory networks. Additionally, movies compared to that was one of the findings, right?
Right.
Compared
so
to resting.
I wish I could show. It is like 3D, like lines which come and goes.
But there is also audio.
There is also music.
Music.
Yeah, like slow music. So it's like a very simple task.
The other question is more general. So many people have observed that during resting state, I'm thinking eyes open is the current standard. Eyes open resting state recording shows the dynamic functional connectivity, shows this coupling, decoupling, or higher correlation in some network followed by another network, and there is a repeated pattern. Now, is there some idea of why this happens during the rest when you are not under any task? Is this some sort of, and so polling visual network, are you okay? Okay, check. Somatomotor network, check.
Right.
Are we doing something like that? Is the brain being in some sort of readiness state, or all parts okay?
I have. So we have shown that the left thalamus, between eyes open versus eyes closed, the left thalamus seems to be involved, but I don't know. Like I have seen what you mentioned about the sequence of occurrences for different ROIs, but I don't really know the underlying mechanism.
Yeah, so the second question was, is there an implication for this? Is the brain doing some sort of, why does this happen, coupling, uncoupling?
I have So I can only tell what I have done. So I have analyzed a lot of data where people have done both eyes open and eyes closed. There's a slight difference in like a very small brain region. I don't see big differences between eyes open versus eyes closed. I prefer to do eyes open because for me, the person not sleeping is very important. And that is why I do eyes open. But others have done eyes closed also. So if you can still stay awake with your eyes closed, I think it is okay.
Right. Okay. Yeah, let's thank Professor Bharat for a wonderful talk. And now hand over to...
Thank you, sir, for such an insightful talk and an engaging discussion. We are sure that students will apply this in their research. And as a small token of gratitude, we'd like to present you with a small memento. Professor Bapi Raju, sir, we would request you to present the memento to Professor Bharat, sir. Thank you, everyone. So we break for tea now, and we meet at 4:45 p.m., that is 15 minutes from now, and on the second floor for the poster session. So the poster session starts at 4:45 p.m. on the second floor, and the GB meeting will be at 6 p.m. in this hall itself, and all the participants can also attend the GB meeting. And a kind request to everybody that please collect your water bottles and don't leave it behind on the chairs. Thank you for your kind cooperation.
Day 2 चाहिए मुझे.
Wow, this is very fun.
Pen drive. Anurag, pen drive. Day 2. Day 2 transferred है.